Kindly join the session fast. We'll be starting the session in 2 minutes. Sir, shall we go ahead? Technical team. I request the technical team to respond.
Yes, ma'am.
Please start recording the session.
Yes.
I request the technical team to give permission to start the session. Dear participants, kindly join the session fast.
Madam, from technical side, it's fine, ma'am, now.
So shall I go ahead?
Yes, ma'am, you can.
Thank you. Sir, shall I start? Professor Raghuram sir. Sir, kindly unmute yourself.
Yeah, thank you.
Sir, shall we start?
Yeah, please.
Yes, sir. Thank you, sir. A very good morning to all the delegates of the Faculty Development Program, sponsored by AICTE ATAL, entitled Internet of Things, Revolutionary Impact on Pharmaceutical Education and Industry. I welcome you all to the Day 2 sessions of the FDP. The schedule of today's sessions are like this. The first session will be handled by Professor A. Raghuram Rao, Pharma Director, Naipar, Sasnagar, and the title of the session is Computer-Aided Drug Design. And the next session will be Challenges for the Adoption of Artificial Intelligence in Pharma Industry by Ms. Kavita, Head Pharma IT Compliance. And the third session will be by Dr. Rajneesh Vasanta, CEO, Andhra IoT, Introduction to Artificial Intelligence and Machine Learning related to Pharma Industry. We request all the participants to join the session fast. Sir, it is my proud privilege to now introduce our today's session speaker, Dr. Raghuram Rao Akinepalli, to the gathering. Sir obtained his postdoctorate from West University, Germany in 1993. Dr. Raghurama Rao sir pursued his PhD in pharmaceutical chemistry from Kakatiya University, 1991. Sir worked as lecturer, assistant professor, and associate professor in the University College of Pharmaceutical Sciences, Kakatiya University, Warangal, India. Professor, University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh, India. At present, Sir is rendering his services as Professor, University College of Pharmaceutical Sciences, Warangal. In his academic exposure, Sir held various positions as visiting professor in University of Minnesota, USA, visiting scientist in University of Bonn, Germany, head Department of Pharmacy in Ministry of Health, Kingdom of Saudi Arabia, DAD Fellow in Westphalisch, Germany. He completed 11 major projects sponsored by UGC, AICTE, CSIR, DST, and one is in progress. Sir had published 19 national and 62 international research papers in reputed journals. He has guided about 16 research scholars and 17 are under progress. His expertise includes design and synthesis of adenosine receptor modulators as anti-asthma agents, aromatase inhibitors, breast cancer therapy. Patents are there, and then impurity profiling and co-crystallization for improved physicochemical profile of poorly soluble drugs is of SAR expertise. SAR collaborated with different reputed institutes like University of Bonn, Saarlands University, University of Würzburg, King's College, London, National Cancer Institute, Bethesda, USA, University of Minnesota. Sir has professional honors and awards include Best Teacher Award by Telangana State Government, Dr. Manjushree Pal Memorial Award for Best Pharmaceutical Scientist of the Year, Career Award for Young Teachers by AICTE, Silver Jubilee Award and Kondaviti Mallayya Shastri Memorial Gold Medal for first rank in M.Pharm by Andhra University. I now welcome Dr. A. Raghuram Rao sir to deliver his lecture. Thank you so much, sir. Now the platform is yours.
Yeah. Thank you very much, Dr. Pawar, for your kind words. I'm very happy to associate myself with this program, a very prestigious program, and very timely also. And at the outset, I thank the authorities of Andhra University, especially the head of department, Pharmaceutical Sciences, and other colleagues and organizers, mainly the convener, Dr. Pawar, for inviting me to share some of my views about the current subject, mainly the Internet of Things, which is revolutionizing several areas of science. And pharmaceutical education, pharmaceutical sciences being one of them. We have no exception from that. And we have several fields which are influenced by the improved developments in the computer sciences, and mainly the everything available online. This has made a lot of revolutions in all these sectors. For instance, the COVID itself has given us enormous opportunities of learning as well as teaching, and in several fields. And of course, research is very much part of it. Well, when I was asked to speak about CAD, I just wanted to give an overview and how several developments, current developments, are influencing or causing so much of change in drug design and discovery. So let me start my presentation.
I request all the participants to be in mute and off your video modes.
Yeah. Is it visible?
Sir, everything is fine, sir. Please go
Okay.
ahead.
Yeah. So before I go to the topic of today's discussion, I would like to uh, pay my respects to the chemists who have really changed the scenario internationally for Indian science. That too, mainly in chemistry, there are certain stalwarts who contributed a lot, and one of them has, in fact, contributed a lot towards drug design and computerization, computer-aided drug design, especially in the protein structure. So let me just give a brief idea about Indian science and chemistry in particular, and chemists who have really contributed from India and made us proud. This is in December 2019, Nature Index has given a overview about the 10 countries which top the ranks in chemistry research, where actually the best chemistry is taking place. This was December 2019. The top 10 countries include China, United States, Germany, Japan, and we are at the 8th rank. And this was the first year where China has superseded USA. And of course, other rankers also there. And we are better than, of course, in this particular thing, from Canada and Spain, among the 10 countries. And all others, including South Korea, are before us. This is rather unfortunate that for the last couple of decades, certain countries have really superseded the Indian science, like Korea. Of course, China has far exceeded in its pursuits. But still, we are still a force to reckon with in an international arena. The one who contributed a lot to drug discovery and medicinal chemistry, basically, is one from our own place, is Elapragada Subbarao garu, who came out with ATP molecule, and he is the one who discovered tetracycline and invented methotrexate. And also, he is known for some of the methods of estimation for amount of phosphorus in the body fluids and so on. He worked in literary laboratories in USA, and a man who is remembered always in the medicinal chemistry research. I was referring to the another gentleman who contributed a lot and who is referred every time when we want to understand the protein structure. That is Dr. Ramachandran, G.N. Ramachandran. In fact, his G.N. Ramachandran plot is very, very popular. And unless we correct it with the Ramachandran plot or obtain a particular protein structure, it can be explained using Ramachandran plot. And he is the founder, he is the pioneer in the field of crystallography, X-ray crystallography. And it became one of the most pioneering fields he developed. And today, most of our molecular modeling and design, they are depending on some of the principles of crystallography and three-dimensional image construction, so on and so forth. Well, we have three more very popular medicinal chemists. It is one, Professor Mansukh Wani. He is the one who gave us both camptothecin as well as toxa, two popularly used drugs, and drugs for as anti-cancer agents. He isolated, characterized, and he is the one who developed, within his lifespan, he has seen that both these molecules, which he and his team isolated and characterized and did clinical trials, everything, and it came into market, and now people are for cancer therapy, these are very popular and used. And we lost Professor Wani very recently. The other gentleman also he lost recently is Professor Harkishan Singh, who developed azosteroids, and his popular molecule chendocuronium is chendonium iodide, is a neuromuscular blocker. And it was also during his lifetime, he has seen that the molecule has been is included in the list of drugs. The other gentleman, again from India, of course, from Andhra University, my own teacher, Professor Venkatrao, he is the one who discovered perovskite and a cardiac glycoside, and this has still been marketed, available in markets as it is as a glycoside. It's a cardiac glycoside. Of course, more recently, we have an Indian-born scientist. Again, he's a molecular biology scientist and became a chemist, and his applications are chemist. And he is from MRC Laboratory, Cambridge, and he is a Nobel laureate in 2009. And more recently, we have still some of our chemists are really doing wonderful job, and they're bringing a lot of laurels. This is one, Thomas John Callaghan. He is one who got one of the prestigious US award in industrial chemistry, and his general manager R&D, Johnson Matthey Incorporation. Well, we have a great past and a great present too. And in the recent years, several molecules are entering from Indian side. You must have heard about it in the recent vaccine research. Several Indian vaccines have already been approved. Not only that, several molecules have been discovered from India. Well, this is mainly because of a very strong chemistry background we have and good application of several components of drug research, and we are getting the benefits out of it now. Well, let's get into the topic of drug discovery being a very lengthy process. And as everybody, one of us know, it is a long and very tedious job, 10 to 15 years, and 2.5 billions, of course, is the cost for a new drug to come. Well, it's a multi-step process. The biggest problem is a multi-step process, which begins with the identification of suitable drug target, validation of the target, hit to lead, discovery, from hits to leads, and then optimization, and preclinical clinical studies. Despite high investments and time incurred for the discovery of new drugs, success rate through clinical trials is very, very, very limited. And there is a high drug attrition rate. Mainly it's because of a long way of from the discovery to filing. So many steps are involved. Isolation, purification, and then identification, preclinical, animal testings and all that, then scale up. And then, of course, it's a further conversion into its development into suitable dosage forms, and then filing an IND, and then human clinical trials, and so on and so forth. Ultimately, it is to be approved by international agencies. It takes a very, very long time. It's a very long process. That's the reason why, where we have this particular process being a very long process, can to some extent reduce the time span with some of the recent developments also. And the pharmaceutical R&D being a very, very multidisciplinary team. In fact, over 100 disciplines have to work together. The moment we are speaking about work together, that means a team, it has to work as a team. And the end result will be really possible and will be successful only when every team member contributes some of them from their whatever they can do. So right from administrative support to animal health, anti-infectious, so bacteriology, so many activities, as you can see, so many fields are involved in this process. And to discover one drug, this is how the pyramidal form of how exactly one drug is coming out of phase 3, phase 4 studies and so on. From more than 10,000 molecules, a molecule may come up as a drug ultimately. So the problem involved in drug discovery is, in the majority of the cases, the drug failure at a later stage has been reported because of lack of optimum pharmacokinetic properties. Its absorption, distribution, metabolism, excretion, and of course, toxicity. Well, you must have heard about recent, our Dr. Reddy's have come out with so many molecules, but ultimately 3 were shortlisted. Ultimately, even one of them was finally has come, but in phase 3 clinical trials, it was to be withdrawn. So that sort of problems are there, where exactly the lack of optimum pharmacokinetic properties are leading to the high attrition rate of drug discovery. But CAD can do some extent. Here exactly CAD can play its role. The use of computer-aided drug discovery techniques in preliminary studies by leading pharmaceutical companies and research groups, it has helped to expedite the drug discovery process and development process by minimizing the costs How can we minimize? Well, to identify the potential hit and make it into a drug lead from lead to hit and failures in the final stage. This is how exactly CAD can contribute. CAD has its ability to contribute in this pattern. The application of rational drug design as an integral part of CAD provides useful insights. Where exactly? The understanding of the binding affinity, how exactly a ligand is binding to the target, and the molecular interactions between the target and the ligand. That is, binding affinity and interaction. Both are important. And CAD can give the true insights, very, very useful insights into this. In addition, the lead identification pharmaceutical research has been facilitated by the availability of supercomputing facility. Well, this is where exactly we have, speaking where we're going up. This is the topic of today's discussion. Supercomputing facility, parallel processing, advanced programs, algorithms, and tools. These are the areas where exactly we are able to get this set of advantage where CAD is contributing in drug discovery process. We'll go by one by one, and let me give a brief idea about CAD, and then we'll proceed to the discussion about how exactly these components are going to contribute. Furthermore, the recent advancements in the artificial intelligence and machine learning methods have greatly aided in the analyzing, learning, and explaining the pharmaceutical related big data. This is where exactly the newer developments, that is artificial intelligence and machine learning methods, are contributing. They are analyzing the big data. They are giving very useful insights and one can learn from that, and explaining every bit of the data in a very, very useful fashion in the drug discovery process. And let me just give you a very brief overview about drug discovery. In this, we have serendipity. Yes, luck factor was playing a very crucial role. Many of our drugs have come out from serendipity. Chemical modifications contributed a lot. Then, of course, screening, very thorough screening of available resources, especially natural products and some of the synthetic molecules. And it has contributed to drug discovery and rational drug design. How exactly we have come into that? We just have a very brief idea about it. The chance favors the prepared mind. We know that. And that's what serendipity says. An accidental discovery. Alexander Fleming, when he studied the contamination of the molds, then he noticed. And penicillin, he has contributed this particular drug. In fact, it is still one of the most non-toxic drugs that we have. Its toxicity is minimal except its hypersensitivity reaction. Still, very, very useful product. Chemical modifications. A lot of approaches for that. In that homologous approach from lead, you could prepare homologous series. Molecular disconnection or simplification, where morphine has contributed. Morphine has been developed up to meprobamate, pentadosine molecules by disconnection approach or simplification of the the pentacyclic molecule of morphine to smaller molecules. That is molecular disconnection approach. It has contributed a lot in drug discovery. Additions also, molecular addition, of course, Nalarfe, is a very popular molecule. And isosteric replacement, so many isosteric replacements, popular among them is sulfatics. And the chemical modification platform, we have several important contributions, like we have cimetidine. It has come out from initially the analog of a known active compound is synthesized with a minor modification, led to improved biological activity. It has come with a very successful development of cimetidine. Of course, a better ranitidine has come out after improvising some of the components of this molecule. Well, chemical modifications, advantages, we end up with something definitely very similar to what you started. That is what exactly. From a reference to a new molecule, we can always get a new molecule, but it's ultimately it is very similar to what we have at the end of once we started. Well, screening, testing a random and large number of different molecules for biological activity reveals their leads. Innovations have led to the automation of synthesis, that is combinatorial synthesis, and high throughput screening. These are some of the methods which have contributed a lot to drug discovery. Of course, the one of the earliest ones was the point to sell to a drug. Well, these of course these are the conventional methods of drug discovery, where we have serendipity or intuition playing a very important role, trial and error approach, and of course it was time consuming because a lot of screening, a lot of tedious procedures were involved, and no new design, and mostly a Me Too approach was responsible for was causing this sort of problems in our drug discovery. Then came the development of rational design. Of course, the example is rational design we have from histamine analogs to the new antihistaminic agents, where we have histamine triggers release of gastric acids. The histamine antagonists to prevent stomach acid release by validating the biological target. And we got the molecules which are developed as guanidine analogs. And in that, a lot of optimization, chemical modifications, lead optimization, and more potent orally active, but thiourea was found to be the problem in the metiamide. Then further improvement was made, and ultimately the ranitidine was developed by chemical modifications. In that, rational drug design played a role. Well, the computer-aided drug design has come into existence initially through quantitative structure-activity relationships. It is QSAR was the first to be developed. Later on, several other things have been developed. Mainly, we have when a protein structure is known, we have two methods. That is known ligands. Then when we know some ligands where the structure of the target is known, we have structure-based drug design methods. And if known ligands are not there, we will go for the de novo design, totally a new design based on the structure of the target protein or target receptor. When we do not have the structure, the unknown protein structure, then we have to go for ligand-based approaches, ligand-based drug design. And here we have similarity searching is there, pharmacophore searching, pharmacophore modeling, and QSAR. These are the common components of this. And CAD of no use when we don't have known ligand and unknown protein. And we have to go for ADMET filters. Let us take up one by one. Let's go to structure-based design approaches. And in that, we have protein ligand docking methods and de novo design methods. Let's see briefly about them. The availability of a three-dimensional structure of a target, therapeutic target, and exploration of the binding site cavities, that is, the active sites, form the basis of structure-based drug design. This approach is specific and effectively fast in the identifying lead molecules and their optimization, which has helped to understand disease at a molecular level. Some of the common methods employed in this are structure-based virtual screening, molecular docking, and molecular dynamic simulations. That means we have several methods in this. Mainly docking is one, structure-based virtual screening, docking, and molecular dynamic simulations. The flowchart is for a structure-based drug design will be protein isolation purification, protein structure elucidation, that is elucidating the protein structure, either by X-ray crystallography, NMR spectroscopy, or homology modeling, identification of target binding site, then molecular docking and structure-based virtual screening. And here, virtual screening of drug-like molecules from chemical databases plays a role. Then, of course, molecular dynamics, dynamic simulation, and binding free energy calculations, and so on. Where then we'll go for the chemical synthesis and biological evaluations, and of course, the molecules, leads which are coming out of structure-based drug design. It examined, once to go for the structure-based drug design, the 3D structure of the biological target has to be examined. And then hopefully one where the target is complex with a small molecule ligand. That is, small molecule ligand usually will be available in a co-crystallized form. That means you get the receptor isolated using a ligand. And unless how the co-crystallized form is isolated, once we take out the ligand, we'll try to understand. We'll understand the active sites. Then look for specific chemical groups that could be part of an attractive interaction between the target ligand and the target protein and the ligand. That's where the active sites are there. And design, based on that, design a new ligand that will have sites of complementary interactions with the biological target. And advantage is visualization allows direct design of molecules. This is where exactly structure-based drug design has played an important role in new drug discovery. In that, the 3D structure of the target is known, and active sites where exactly the ligand is interacting with the target is understood. And based on that, the visualization of the ligand and target interaction will help us in understanding how exactly we can design a new molecule. In fact, this is where exactly in all these aspects, that is purification, understanding the structure of protein at very macromolecule, and understanding the active site, we have the new methods, that is the advances in the computerization, that is in the IoT, Internet of Things, and the machine learning methods are helping us to understand this better. Well, based on this, we have docking. We put a molecule in the approximate area where exactly binding is taking place. And docking algorithms encodes orientation of compound and its conformations and optimize binding of the ligand to a protein with minimization of energy and interactions to use hydrogen bonding interactions and maybe some hydrophobic interactions. Based on this, you can get the functions called scoring functions. Based on that, we may be able to identify which may be the actual, maybe a more suitable ligand. And we have a protein here, and its binding site is understood. Then we try to develop a lead, a ligand molecule, a small molecule. As you can see, protein is a macromolecule. To understand the active site, where exactly the binding is taking place, understand this active site is a complex process. And how exactly we can dock this molecule, a new molecule, or maybe a known molecule, and we think that it is going to interact with that. And the interaction is leading to a ligand and target complex, and which is responsible for a biological effect. So this is where exactly docking plays an important role. A structure means using a protein structure, computational method that mimics the binding of a ligand to a protein, and that leads to an interaction between a ligand and a target. This can predict actually the pose of the molecule in the binding site, the binding affinity of or a score representing the strength of binding. This is where we can understand how exactly the molecule, the ligand, is binding, and how exactly the binding affinity, how strongly it is interacting, and what is the binding affinity based on the score. So this is how the structure-based drug design, where we use docking as a protocol for understanding or developing a new molecule. And how exactly the binding site is understood? Identification of the ligand binding site. This is the crux of the issue. The information about ligand binding and its site is a prerequisite for carrying out specific docking. The knowledge of the binding sites can be extracted from the site-directed mutagenesis study or X-ray crystallographic structures of proteins co-crystallized with substrates or inhibitors. This is the key for the understanding ligand binding interaction. While the experimental information about the binding site of many proteins is not available, this is the crux of the issue. When we do not know the experimental information about the binding site of many proteins or many targets, there is plenty of software and web servers which can help us in understanding predicting the putative binding sites, the proteins. See, we have plenty of web servers available to understand this, because of the problem is the experimental information about the binding site is very difficult to get. So we have plenty of software, several software programs available, web servers available, like CASTP, Docksite Scorer, Nsite Match, Depth, MS Pocket, Metal Pocket, and QSite Finder. So many such programs are available, which are giving us an idea about the ligand binding site, identification of the ligand binding site. This is what for instance, several years ago, 5 years before, such programs were not available. And today, with the invention, with the development of so many software programs, we are able to understand or predict the putative binding sites of the target proteins. The bulky compounds which do not fit well within the binding site are rejected during the lead identification procedure. This is where we can identify the one which is not fitting into this. And probably we can reduce the chance factor by understanding the ligand target interaction, especially the ligand binding site. So this is where exactly CAD is contributing a lot. And the developments of the newer developments are going to affect or going to give us some insight into these problems associated with the new drug discovery. Well, the next part is molecular dynamic simulations. The simulations, molecular dynamic simulations of protein was first performed way back in 70s. This powerful physical technique was used to predict the positions of each atom in a molecular system with respect to time, which is based on Newton's laws of motion and governing interatomic interactions. So this is a well-known concept. The forces between interacting atoms are estimated using a suitable force field, which is used to determine the overall energy of the system. MD simulations have been widely used for several reasons, especially the position and motion of every atom of the system are captured at every point in time, which is quite tough using any experimental technique. This is where exactly another issue is. That is, experimentally, it is very difficult to understand the position and motion of every atom in a particular system, especially when we are dealing with a macromolecular body. These simulation conditions are exactly known and can carefully modulated also using these MD simulation techniques. Again, in the structure-based drug design, these are very popularly used, as this technique helps to unravel many atomistic details about binding, unbinding, and conformational changes in the receptor at a fine resolution. This is very, very important. At what resolution we are trying to understand the details. And that too about the binding, its unbinding and conformational changes. To understand that, we need a very powerful programs which cannot be obtained by experimental studies. This is where exactly the programs, software programs, mainly the newer developments are going to help us. And of course, some of the programs are earlier known like AMBER and CHARM, but some other programs are coming up and several more are coming up. So I'm just putting a few of them. And this is going to give us a very useful insight into the ligand target interactions. If this is the structure-based drug design, which is being contributed, it has contributed a lot for the development of a large number of new drugs. In fact, I am not giving the list. In fact, a very long list is available. And about what are the success stories of each method that we have in the CAD. But a lot of literature is available about them. And today, it has become very, very common in all the pharmaceutical industry to use CAD as one of the most important methods of new drug discovery. Well, the other one is de novo, totally a new drug design. That's how exactly we design a new drug based on some of the data available, some of the ideas, some of the rather intuitionally, some of the information that is available. It builds compounds that are complementary to the target binding site on a protein via a random combination of small molecular fragments. That means no ligands are available. Then you may have to design new molecules totally, that is in a de novo fashion, that is totally new design. And there are so many examples for this. For instance, taking a fragment, it's based on fragment-based approach, that is taking a core fragment and trying to develop into, you add some more a core molecule, a core moiety, and add some of the fragments, and ultimately you can have their different conformations also, and maybe you are leading to a new molecule, you design a new molecule with an intention that it is acting as a complementary to the target binding site. There are several such examples in literature about de novo drug design, where new molecules have been rather tailor-made to a particular receptor. Well, if these two are the structure-based methods, that is, we have where the protein or the target molecule target receptor structure is known, we go for the structure-based design as well as our de novo drug design methods. The other popularly used method is ligand-based, where the structure is not known. It is another widely used approach and is employed when three-dimensional structure of the target is not available. This is the issue with most of the targets whose three-dimensional structures are not known. Maybe known to some extent, but at what details we know it is very, very difficult. And most of the times this is the situation. The information derived from a set of active compounds, then you have to go for ligand-based only. That means a known ligands which are active or which are shown to interact with a protein, with a target. But the target structure we don't know, but ligands are active. So it implies that the ligand is acting on the target and leading to a benefit, to a therapeutic effect. So based on that, the design is taking place. The information derived from a set of active compounds, again, is a specific target receptor, can be used in the identification of physicochemical and structural properties responsible for the given biological activity, which is based on the fact that structural similarities correspond to similar biological functions. This is the assumption. This is the structural similarities correspond to biological functions. See, some of the common techniques in this is in this particular ligand-based screening approaches, developed drug discovery design methods. We have pharmacophore modeling and quantitative structure activity relationships and artificial intelligence. These are the three ways by which we are able to develop new drugs, where exactly the artificial intelligence has been added recently. And this is contributing a lot to some of the discoveries which are taking place. Well, let's take one by one. Pharmacophore modeling. The pharmacophore is a spatial orientation of various functional groups or features in a three-dimensional manner necessary to show biological activity. And in the pharmacophore models, again, we have distance geometry-based qualitative common feature hypothesis is one, and quantitative prediction pharmacophores from a training set with known biological activities. That is, there are two types of models one can develop: distance geometry-based approach and quantitative prediction approach using a training set. So this is how exactly ligand-based drug design can go for. To some distinct usages, what is a pharmacophore? the substructure of a molecule that is responsible for its pharmacological activity. That is something like a chromophore. The part of the molecule which is responsible for a pharmacological activity, we are calling it as a pharmacophore. A set of geometrical constraints between specific functional groups that enable a molecule to have biological activity. That means, see, a hydrophobic area and a hydrophilic area, and they are connected with a chain of with a carbon chain of 3 or 4, and then an aromatic side, something like that. You have some sort of geographical or the orientation of geometrical constraints between different functional groups, leading to a particular hypothetical pharmacophore that we can create, and we can understand it. Through that, we can start working on such a pharmacophore. And from activity data, we generate a pharmacophore basically. This is from the activity data into known ligands, we generate a pharmacophore. Basing on the pharmacophore, search in a compound library, a large number of compound libraries. In fact, lot of commercial as well as freely available open source chemical databases are available, and screen them using this type of biological, this type of pharmacophore, and then this from this you can, that is the chemical database can be purchased or synthesized also. You can have your own and test their activity, again contribute and improvise the pharmacophore and again go on. This is an iterative process, and this iterative process can yield some of the very potent molecules. And it examines both active and inactive molecules, and based on that, generate a hypothesis about which chemical groups on the ligand are essential for biological function and which chemical groups suppress the biological function, that is active and inactive. Both of the groups, both of the components we try to understand. Based on that, generate new ligands which have the same and necessary chemical groups in the same three-dimensional locations. So this is how a new design based on a pharmacophore modeling can lead to a lot of molecules, a lot of new molecules. The advantage here is we don't need the biological target structure. That is, the target structure is not required. We have developed a pharmacophore model. Based on the model, we try to screen the molecules. Here again, we have some steps: training set selection and features selection, conformation generation, and then common features, and of course, validation. You have to validate the pharmacophore. Of course, these have to be followed. These have to be followed in a very meticulous manner, because training set selection, that means you need to have actives and inactives, and in that which feature is active and which feature is leading to inactive one, we have to understand. And based on that, a pharmacophore model has to be generated. Here, some assumptions are there, considerations or assumptions. Training set molecules, diverse in nature, contain maximum structural information, and most potent within the sets. So, as you can see, this is a very, very critical step in pharmacophore modeling. And here again, exactly two things are important. One is what type of biological data we already have. Remember, we are dealing with the ligand-based approach. In this, you need to know which are a very, very valuable biological information has to be available, where the structure has to be a diverse set of structures we must have, and then structural information must be appropriate, and we must know which is the most potent among the series and which is the least potent. Features should be selected on the basis of SAR studies of the training set. And each training set molecule should be represented by a set of low energy conformations. And conformation generation technique ensures broad coverage of conformational space. And align the active conformations on the training set molecules to find the best overlay of the corresponding features and judge by statistical profile and visual inspection of the model. So these are some of the very, very crucial steps involved in the pharmacophore model generation. And here again, to take to understand a big data, the newer methods of IoT and machine learning methods are giving us enough or a very, very valuable information about it. And what are the pharmacophore features? Hydrogen bond acceptors, hydrogen bond donors, hydrophobic sites, hydrophobic aliphatic as well as aromatic aliphatic sites, and positive charge ionizable, positive charge, negative charges ionizable, and rings, aromatic rings, aliphatic chains, and so on and so forth. And each feature consists of, again, its chemical function, location and orientation in the 3D space, and tolerance in location, and of course its weight, its mass. So these features of all these parameters have to be made available, become part of our pharmacophore generation. We have input structures, conformation modeling, SAR data, we generate a model and evaluate the hypothesis, and based on that, once we evaluate in sense, once we make it into a standard model, validate it, then it becomes an acceptable pharmacophore. So these are hydrophobic, as I told you, negative ionizations, positive ionizations, and ring and aliphatic groups, and hydrophobic aliphatic, ring aromatic aliphatic, and so on and so forth, leading to a pharmacophore that is developed as a hypothesis which is to be mapped over the active molecule. And ligands having different scaffolds, but similar spatial arrangement of key interacting functional moieties can be identified using the pharmacophore-based virtual screening. Here again, the bioactive conformation of the molecules within the target binding site can be incorporated in the pharmacophore model. The pharmacophore model is also often used in QSAR studies in the molecular alignment stage. Some frequently used programs include Catalyst, Phase, Ligand Scout, Galahad, and PharMapper. A good pharmacophore model also incorporates spatial constraints in regions occupied by inactive molecules and often optimized further to make the model less restrictive. All the pharmacophoric features which are not consistently detected in an active molecule are either made optional or removed from the final model. So this is an advantage in pharmacophore-based modeling. Well, the other one, which is rather became conventional, I mean to say, it is one of the oldest. In fact, all these methods have been developed later. The first one was QSAR. QSAR certainly has contributed a lot in the new drug discovery. And this is another ligand-based approach because we are understanding the active and inactive ligands. And based on that, we are trying to understand which particular biological, which particular part, which particular aspect has to be understood, can be understood basing on their physicochemical parameters. QSAR studies are based on the principle that variations in the biological activity of the compounds can be correlated with the changes in the molecular structure. They are widely used in the drug discovery process, in the hit-to-lead identification, or very importantly, we use the term lead optimization. A statistical model basically, which is constructed using these correlation studies, and the final model can be used to predict the biological activity of a new molecule. The key requirements in the generation of reliable QSAR model, again, here a model has to be developed. How a model is developed? Based on a sufficient number of data sets with biological activities obtained from common experimental protocols. That means first the biological data has to be available. Then the training set and test set molecules must be appropriately selected. This is one very, very critical aspect in QSAR, where the training set and test set, data sets and training set has to be very, very carefully selected. New autocorrelation among the physicochemical properties of the ligands that may cause overfitting of the data, and the applicability and predictability of the final model must be checked using internal and external validation methods. This is very, very crucial for the QSAR model to develop. Again, we have assumptions. What are the assumptions? The effect is produced by model compound and not by its metabolites. This is a very, very important assumption. QSAR just believes that it is just by the compound, but we don't have any idea about how and when, if and when a metabolite is active or more active than the compound itself, the molecule itself. The proposed conformation is the bioactive one. This is where exactly a crucial decision has to be taken. That means assumption basically. The one that is conformation that we have developed or we are proposing, it is the most active post conformation. The binding site is same for all the model compounds. This is an assumption again. The binding site is the same for all the model molecules. The bioactivity explains the direct interactions of the molecule and target. The biological activity will explain the direct interactions of a molecule and a target. This is also an assumption. Then pharmacokinetic aspects like solvent effects, diffusion effects, transportation, that means its reaching of the target, are not under any consideration. This is, unfortunately, these are some of the assumptions in QSAR. That's the reason why, although QSAR has been one of the oldest, rather, among these methods that we have just discussed, but still, these assumptions have to be taken forward. And in all these, of course, again, these the newer methods are giving a better insight into these assumptions. QSAR generation process, as we have already seen, selection of training set, entering biological activity data, generate conformations, calculate descriptors, large number of descriptors again available, selection of statistical method that we are going to use it, and generate an equation and validate an equation and predict for an unknown. So these are several steps involved in QSAR process, QSAR modeling, QSAR-based, ligand-based drug discovery, drug design. Again here, descriptors. How many descriptors? Which descriptors are biologically active is a very difficult, very, very important thing. And of course, how many types of descriptors? Structural descriptors, electronic descriptors, quantum mechanical descriptors, thermodynamic descriptors, shape, spatial, conformational descriptors, and of course, last but not least, receptor descriptors. So many descriptors are, in fact, we have a very large number of descriptors are given in literature. And that is true also because so many descriptors will certainly, each one will influence the biological activity. For that, selection of descriptor is again a very important component. What is particularly relevant to the therapeutic target? That is, which type of descriptors are relevant to the therapeutic target, the target? What variation is relevant to the compound series? What type of variation is required in a compound series? What property data can be readily measured? Which property data you can easily measure? What can be readily calculated? All of them have to be very important decisions one has to take. And this is the tough part of QSAR study. And of course, we have a large number of 3D QSAR program people have worked. In fact, I'm just listing out some of these programs, and there are linear methods, non-linear methods, and several three-dimensional QSAR programs are well known, and each of them has really contributed to the DUSG story so far, and they are being improvised using some of these programs, some of the drug discovery programs. Hypogen module of catalyst, phase, COMFA, COMSIA, so many are the 3D QSAR programs. And they can be the QSAR techniques can be classified into linear and non-linear based on the chemometric methods that we use. And here again, the linear methods include linear regression, multiple linear regression, partial least square method, principal component analysis, and principal component regression, and all these techniques are there in the QSAR. The examples of nonlinear includes k-nearest neighbors, artificial neural networks, and Bayesian neural networks. These are the nonlinear methods, descriptor, the chemometric methods that we use in QSAR programs. Then which can be pursued? One can pursue both structure-based approaches as well as ligand-based approaches independently, and one can pick up the best out of both, and probably one can design a new molecule based on this. If the binding mode of the ligand and target is known, information from each approach can be used to help each other. Let us remember one thing. All these methods are complementary. One has to understand like that. And ideally, identify a structural model that explains the biological activities of the known molecule, known small molecule, on the basis of the interaction with the 3D structure of the target protein. So you know based on the receptor that is structure based, you have a pharmacophore model, and then probably once we know this particular design is possible, then you can go for a new molecule having that component as part of the molecule. Pharmacophore based drug design will give you something about molecular mimicry. That is, how exactly two molecules are mimicking each other. A model and a known ligand. This is how exactly a pharmacophore based approaches. And the structure-based approach, this is a ligand-based approach. We have pharmacophore developed, and that molecular mimicry may be understood because based on the pharmacophore, we design a molecule. We can see that how exactly they are mimicking. How exactly one hand is mimicking the other. But in the In the structure-based drug design, that is receptor, when we have the structure of the receptor, based on that, complementarity is understood. That is the mimicry and complementarity. Although they look similar, they are not similar, they are not same. And which one actually, both are useful or both are valuable, but which one actually fits into the assumption that this is the actual potentially active molecule? How do we understand that? We understand based on the biological data. Typical projects are not purely structure-based or ligand-based approaches. They use combination of both. And hopefully, synergistically, one has to use them. And in all this, a typical project may be between something between these two. Pure pharmacopoeial structure-based, ligand-based approach here, and pure structure-based approach. The typical project may be in between, but the best one will be, ideal one be, the one which combines both of them in the best possible way, and you get a successful product out of that. And how exactly the artificial intelligence is going to, what we have seen under ligand-based approaches, we have named as, first one is pharmacophore based design, that is one of the ligand-based approach. The other one is a conventional QSAR approach. And thirdly, we have the artificial intelligence methods. Artificial intelligence is a type of machine intelligence that relies on the ability of computers to learn from the existing data. I am not going, this seminar itself is going to speak a lot. So many speakers are going to speak about artificial intelligence and the Internet of Things and revolutionary changes they are bringing about in every sphere of scientific discoveries. And drug discovery is no exception from that. And several more speakers are going to give more insight about artificial intelligence. And let me just briefly touch upon how exactly this is going to help us in drug discovery and how it is being currently used. It is the ability of the computers to learn from the existing data. The problem is we have a huge data. To understand, analyze the data, give an essence out of the data, giving the most crucial information for a new drug design, artificial intelligence is going to play very, very important role. Artificial intelligence is being used in various computational modeling methods to predict the biological activities and toxicities of drug molecules. See, activity, of course, is very, very important. But what is even more important is the safety. So toxicity, free from any toxic effects. So in both of these things, activity, that is biological activity, and its unwanted side effect profile or toxic profile, understanding both of them, artificial intelligence is going to help us a lot. Further, it has very wide applications in understanding, well, prediction of protein folding. Well, what we have seen so far is the target structure, and we are considering it as a static one. But unfortunately, in the biological system, nothing is static. It changes. And how it changes? The proteins or the macromolecules, which are the most common targets, they have properties like folding. And what about the protein-protein interactions? Prediction of protein folding. Artificial intelligence can give us very important insight from that. Protein-protein interaction, virtual screening. Well, this is a very commonly used term. That is in silico screening. In silico screening or virtual screening, artificial intelligence is going to give us much, much better idea from that or results from that. As I told you, QSR itself, so many assumptions, and all those assumptions can be understood with a fine details or micro details based on the application of artificial intelligence. Evaluation of admit properties. What we have seen, most of the drug discovery or new drug discovery is getting a problem because they are failing at the admit properties: absorption, distribution, metabolism, excretion, and toxicity. These are the 5 important components of a molecule to be considered as a drug. For that, to understand that, the admit profile of a molecule, one can have a better understanding using artificial intelligence. Of course, not least, it is de novo drug design, new drug design, which is another one where we do not have any ligand. But the structure is known. That's what the problem with the large number of data that is available about some of the molecule structures we know, but there is no ligand exists, where we have to go for de novo drug design methods only. In all these aspects, artificial intelligence is going to play a crucial role, is already playing a role, and it is going to have much, much better or more important contribution in the years to come. In continue, there are 2 powerful methods in this. In this rational drug discovery, that is in the computer-aided drug discovery programs, machine learning and deep learning. I'm just giving an overview about this. But these 2, I think these are the 3 components: artificial intelligence in that, machine learning methods and deep learning methods. This particular entire session, several speakers who are specialists in this will be putting a lot of light over these things, where we have machine learning algorithms that have been extensively used in drug discovery include support vector machine, SVMs, random forest, and Bayesian methods. They are machine learning algorithms which are very useful in drug discovery. And in the deep learning, machine learning and deep learning, and in the deep learning processes, we have convolutional neural networks, deep neural networks, recurrent neural networks, autoencoder, and restricted Boltzmann machine. These are the components. These are the different methods that we use in the deep learning process, which are part of the artificial intelligence. That means we have both machine learning techniques as well as deep learning methods, and in that the algorithms are different, and each one of them is going to contribute a lot to the drug discovery process. Just to share some of our work in this, in the development of a new drug based on 2 approaches, that is pharmacophore based and docking based 3D QSAR methods. We try to develop some new molecules, and our experience on non-steroidal aromatase inhibitors, I would just like to share with a couple of slides how exactly we could get some sort of insight into this particular development of new non-steroidal aromatase inhibitors. where we have developed based on pharmacophore mapping and docking-based QSAR. New molecules were developed, and here we have pharmacophore modeling studies were performed to understand the spatial arrangement of pharmacophore features, and then docking-based QSARs were performed to develop the QSAR model, to develop good, better scores for the descriptors. And we have designed based on a pharmacophore model. We have developed way back in 2006 when the structure was not known. The target was aromatase enzyme structure was not known. We developed a model, and later on in 2009, the actual crystal structure of the placental microsomal aromatase was known. But luckily, our homology model was almost very much, or more than 95% matching with the actual structure, and thereby we were successful in developing some molecules, and which were found to be very active, biologically active, and some molecules we developed, and some of the molecules we patented also, and those patented molecules were, the technology was transferred to some of the industries, and they were developed to some extent, and once they are developed and their toxicity was studied and everything was done, but only clinical studies phase was due, and at that point the decision of whether to go ahead or to wait for some time or keep it as a reference molecule or something like that, it's a commercial decision, and that is how we ended up with some of the new molecules whose patents we could transfer to an industry, and we got some success in developing new non-steroidal anti-aromatase inhibitors for breast cancer therapy. Finally, I would like to, coming to the end of my talk almost, that is, I have a piece of advice to my colleagues who are pursuing medicinal chemistry, and today a lot of programs, software, open source software is available for prediction of activity and designing of new molecules. For that, I have a piece of advice to my colleagues and researchers. First and foremost is, for a medicinal chemist to be successful, good organic chemistry is essential as a basis. Of course, as all of you agree with me, this is one. And the critical view of the methodologies need to be understood, practiced. The feasibility parameters, that's about, I'm speaking about organic chemistry, of that too, preparative organic chemistry. We use this term, preparative, in the sense we are going to make them in larger scale. In that sense, the parameters like conditions, reaction conditions, percentage yields, formation of related products, purity challenges, especially in the step of purification, etc., need to be addressed on a regular basis. The disposal of solvents and sewage management, which is a green chemistry aspect, has to be taken care of, because this is, however active a molecule may be, unless these things are understood in a proper way, it is very difficult to proceed to become a successful molecule. Then the most important, once we are developing based on especially some of the software which is available and which is going to help us a lot in developing a new designing a new molecule, what is most important is how exactly you are understanding the target and ligand interactions. This is where the crucial step is this, that is, target we know, and how your ligand is having some interaction with that. And what type of interaction, one must have a clear idea about this, because all the other things will follow. Computational methods are good aids in analyzing large data. The key is the credibility of the data. This is where exactly I would like to caution. one has to be very, very careful in taking or selecting the biological data which is available in the sense which is already available to publications or so on. So how credible the data is, because all other assumptions, all other things are based on that credibility of the data that is available. So unless you are sure about the credibility of data in the sense the molecule, the people, the researchers who have presented that particular biological activity, how much they cared about the purification of the ligand, of the molecule, of the chemical that they have used, or the molecule they have used, and what type of biological model they have used, and what is the data that they have presented, how it was standardized, how it was compared with the standard molecule, whether the standard molecules activity is already reported or already known, and how best they are getting correlated with each other. So these things are very, very important. And realistic approach and ethical reporting will improve the credibility. The credibility of the biological data, credibility of research data is always based on the ethical and realistic approach of the researchers. There is another important thing. Negative results need to be appreciated. Currently, a large number of good journals are available which dedicate negative results. That is also very important. We try to understand, we expect something, we get a data, it's fine. But sometimes we don't get it. That is, sometimes we never know our model was good, our everything was good, but still it biologically it was not active. So that's a negative result. So for such negative results also have to be reported, and they also have value once we are predicting or once we are developing a new model. And as I told you, one very, very important thing, computational methods are good aids in analyzing the large data. But the ultimate decision has to be taken by the medicine chemist, who has to look into each component and how best a particular component is going to help us, how best we are going to take up a particular descriptor, how best we are understanding, and how much statistics is being applied in taking a decision and all that. It is very, very crucial in designing a new drug. And today, it's an advantage as well as disadvantage. A number of programs are available, just online programs, software programs are available, easily accessible. Sometimes we will be carried away by some of one may be carried away by some results, which are sometimes very fascinating, but they are to be actually validated. Unless you validate, you are not sure about what exactly the results that you got. And unless that validated approach is used, your data cannot be credible. Unless the credible data is there, a better model cannot be designed, irrespective of how much of our software programs are offering us. With this caution, I would like to thank every one of you for making making me to reach every one of you through this forum. And I could share some of the information, some of the things about the new developments in the artificial intelligence and machine learning methods that are contributing to drug design. And I would like to suggest 3 very recent publications which are available online, and please go through these publications. You can have better insight into the topic that we discussed today. I thank all the audience, and this particular presentation I would like to dedicate to the great teachers who trained during my course of my studies, and all the students who really participated, contributed immensely by working with me in my research group. Thank you very much for your attention.
Thank you, sir.
Yeah.
Sir, here we have a couple of the questions in the chat box.
Yeah. Okay. Okay. Yeah.
Shall I read out, sir?
Yeah, I'm going through.
Yes, sir.
How can artificial intelligence helpful predicting drug toxicity? One question from Tadesi. Tarikin Tadesi. It's a very good question. As I mentioned already, lot of toxicity data is available, but it's very difficult to analyze because the data that we are getting in through various sources, uh, artificial intelligence is helping us because all this, the artificial intelligence is a good procedure, good method to analyze the data. So far, we could not get good access to the large data, especially when we do the phase 3 clinical trials and phase 4 clinical trials. Phase 4, of course, post marketing. Phase 2 and phase 3, where we get a lot of toxicity data, and a lot of data includes a lot of toxicity that can be analyzed better using artificial intelligence tools. So this is going to be a revolution in the drug discovery, where drug discovery in the sense drug development of a new molecule as a non-toxic material. I can only give up to this, and this is already being practiced. Well, whether any data on drug discovery available online for us to work with algorithms to get some experience? Yes, there are. In fact, lot of data is available in variety of websites. You can just access the drug discovery data on drug discovery. Drug discovery in the sense, of course, the data available from companies sometimes is very difficult to get, but there are some open sources. You must have heard about open sources of drug discovery, OSDD programs. In that, lot of data is available, and it is available online, and you can apply the algorithms to get some experience. That's good. Good question. Sai Padmi sir, can we predict agonism and antagonism by docking studies? Yes, of course. Yes, we can do it. Antagonism, yeah, we are one cap. Bansal, Vijay Kumar Bansal, what is retrosynthesis? Wow, good. Where retrosynthesis approaches or taking a, you know how exactly we know a ligand which is active. That ligand you want to synthesize. So retrosynthesis is you using how exactly you can develop that molecule. You know the target, and you break it up into its components. How best you can break it up into components, and each component, how can you join them? This is what retrosynthesis is. And in that, several procedures, one has to understand, there are of course so many techniques in that again, and the retrosynthetic approach only one has to develop. If a known molecule is there, you can develop several methods of synthesis using different components of its fragments, and each fragment can be synthesized separately, and those fragments will be clubbed or compounded to get your molecule. Indira Devi, could you please explain that the methods to predict binding cavity size of an enzyme or receptor? Yeah, active site, to understand the active site. I have already given you, in the presentation is available, and in that we have given, and the ones that I have mentioned in the suggested reading, there are plenty of methods which are given, plenty of servers which are given, which will predict, which can explain the prediction of binding cavity. Prediction of binding cavity incidence. Binding cavity incidence is the one which is active, and that has to be understood. That has to be taken in. That can be understood. That can be predicted. The active site can be predicted using these a large number of programs available, and most of the programs are online programs. One can see, I have given a list of molecules, list of these things. If you want, you can go back also. I have given that where exactly the active site can be understood. And I have given that. Please go through. They are all open sources. So, Divya, what are the top rated journals in our pharmacy field? The top rated journals in our pharmacy field, right? There are several journals. Please don't think that the journals, there are plenty of journals available. And please remember, while understanding the top rate, you said top rated. Top rated rating of a journal is based on its impact factor. People use one term called impact factor. Impact factor in the sense how much it is. It's basing on its readership. How many people are referring it, and like that. Based on that, you will understand. So, if the high impact journals, or there are, in fact, if you consider impact factor of some journals, and say for example, Journal of Medicinal Chemistry, Journal of Pharmaceutical Sciences, their impact factor is really not very high because it is only read by a part of some specialist. Only pharmacy people or medicinal chemists will read. If you compare the Journal of Organic Chemistry or Journal of American Chemical Society, like that, the impact factor is no way comparable. But within the pharmacy field, some of the journals like Journal of Pharmaceutical Sciences, Journal of Medicinal Chemistry, European Journal of Medicinal Chemistry, European Journal of Pharmaceutical Sciences, like that, and even bioorganic and medicinal chemistry letters, bioorganic and so like that. There are plenty very good journals, and for Journal of Pharmacy Pharmacology, like that. There are good journals available, and unfortunately, for some of them are not easily accessible. Also, that means the full papers are not easily readable, not easily accessible openly. But today, a lot of open access journals are also coming, which are being available and which one can go through. But publishing in those journals also is becoming difficult, some of the journals, because there is a lot of fee, high amount of publication charges are also there. But still, there are pros and cons. There are certain journals which freely they publish. One can target them and publish. Yeah. Thank you. Any more questions?
Sir?
Am I audible?
Yes,
Yeah, please. Yeah, Jagdish.
Sir, actually, in all the 4 approaches, we have got something which are missing the real active drug. Okay. Because the recent concept of induced fit theory, and in the design, so the protein is inducing itself depending upon the ligand properties. So in that case, we are all the 4 approaches leaving that active moiety and hitting to something else. So what is the better approach to take? That one is actually a drug and we are leaving it now. Is there any approach?
Yes, so really good question. Yes, today, why induced fit is a well-known concept basically. I was referring to while going through some of the approach about the structure-based thing, a form of structure-based. We are saying that the protein or the target is a static one, which is actually not. I mentioned that also. Yeah. Am I audible to you?
Yes, sir.
Yeah. Mr. Jagdish, your question is valid because the assumption One assumption is our target is a static one, which is not actually. So far, all these programs, all these all these programs, all these methods were developed based on certain assumptions, and that assumption is always is not correct. As you yourself mentioned, the assumption is not correct because the protein is not correct. But now, with the artificial intelligence is giving us some clue about protein folding and insight into the drug receptor interaction, right? Which were not available earlier. This is where exactly a key development is expected to take place, or the development which is expected to take place with regard to the development of your question. I certainly agree with you. I agree with you. But the artificial intelligence approaches, methods are going to help us certainly in understanding this.
Thank you, sir.
Yeah.
You made the topic so simple that anybody can understand, even layman also. Thank you, sir.
No, not that. Please don't say that. But it is, unless we make it, unless we understand it in a simple way, it's very difficult. Otherwise, so many programs are available, so many things are available, which is rather confusing today. Right? Thank you.
Thank you, sir.
I request the participants to kindly clarify your doubts if you have any. You can straight away ask, or you can post your queries in the chat box.
Yeah, you can post your questions. You can send to the convener, and which I will certainly give any information. I already, as I mentioned, the suggestive reading which I mentioned, 3 articles, they are openly available online. That's why I am not provided. Please go through them. They certainly clarify some of these pertinent questions.
Sir, it was an amazing lecture. Dear Professor Raghuram Rao, sir, we are very, very happy to hear about the bridging of updates in computer-aided drug design. Your lecture boosted our knowledge and was very fruitful to all the participants. In your lecture, you discussed on various aspects of computer-aided drug design, like contribution of eminent scientists in drug design, discovery, and development, various disciplines working together for drug discovery, problems of drug discovery, computer-aided drug design in drug discovery, various approaches to the drug discovery, mechanisms of structure and ligand-based drug designs, pharmacophore modeling, overview, generation and hypothesis, quantitative structure activity relationships, QSAR, artificial intelligence and drug discovery and its applications. You advised the chemists, you suggested the reading of latest references. Thank you so much for accepting our invitation and throwing light on us on the topic computer-aided drug design from the beginning of its history to the latest readings. Thank you so much, sir. Namaste. Thank you so much for taking a lot of pain in enlightening us, though the time there is almost, I think, midnight. Thank you so much, sir.
Thank you so much, and thank you for all of you for arranging and your computer team and your everybody cooperated. I'm so happy. Thank you very much. And any more questions, if you get, you please kindly pass on to me, and I'll be trying to give whatever is possible from my side. Thank you so much. Thank you, everyone. Namaste.
Thank you, sir. Namaste. I now hand over the mic to the technical people for the further proceedings.
Yes, ma'am. I request all the participants to come to video mode so that we can. I request all the participants to come into the video mode.
Yep.
I request all the participants. Sir, I request Raghuram Rao sir also to be.
Video mode? Okay. Okay. Yeah. Is it
Sir,
okay?
yes, sir. Sir, you
Okay.
can close your session.
Oh, okay. Yeah, sure. Yeah. Okay.
We still have some participants in off mode. We request all the participants kindly come into your video mode so that we can conclude a session with the topic. All the
I
participants,
request Anita Singh.
Dinesh sir, Mrs. D. Nagraj, Anand, Kiran Bala B, Deepak Tomar, Ankita ma'am, Madhura Dikshit, Simran, Jogendra sir, Ravindra Babu sir, Rama Devi ma'am. I request all the participants for just 1 minute.
Okay.
And here goes 3, 2, 1. Yes, ma'am. Thank you. Thank you, everyone.
Thank you, sir. Thank you so much. Thank you, everyone. We again join the session at sharp 11:25.
Okay.
Thank you, sir. Now I hand over the session to the technical people.
Yes, ma'am. So I request all the participants, you can, ma'am, do we proceed to a break now?
Yes, but before that, you announce the formal instructions to the delegates.
Yes, ma'am. We request all the participants to kindly note some general instructions. All the participants are required to join the technical sessions of 5-day faculty development program prior to the session start, 10 minutes before into the session so that we can sort out any of the technical issues before the speaker is starting his or her presentation. The next point to be noted is we request all the participants to attend all the sessions of 5-day program because the attendance of the participants is to be uploaded to the AICTE portal, and the minimum requirement of attendance which is suggested by the AICTE authorities is 80% of minimum attendance to generate your certificate. And along with that, a score minimum of 60% marks in the test, which is to be conducted at the end of the program, that is after the completion of all the sessions. So we request all the participants to kindly note these 2 points because later on, these 2 points are taken into consideration for the generation of certificate, noting the completion of faculty development program from AICTE. Thank you. Thank you, ma'am. Over to you.
Thank you, dear technical team. So let's have a small break for about 25 minutes and we'll be back at sharp 11:25.
Kindly note, Mr. Bommepalli Nagarjuna. Sir, you are presenting your screen. Can you stop the presentation? Mr. Bommepalli Nagarjuna. Yes, thank you, sir. Good morning,
Good
Kavita.
morning, ma'am. I hand over the session to the technical people to help the speaker.
Yeah, now can you hear me, ma'am?
Yes, ma'am, you were audible.
Thank you.
Kavita, ma'am, can we check with your presentation? Like, are you able to share or not? Once we'll check, ma'am,
Yeah, sure.
before we start. Yes, ma'am. Yes, ma'am. It's fine, ma'am.
Yeah, thank you.
Thank you.
Madam, everything is ready. We'll be waiting for 5 more minutes to join the rest of the participants. Three. technical session.
Yes, ma'am.
Your team is ready.
Yes, ma'am.
Shall we proceed?
Yes, ma'am, you can.
Yes. Ma'am, shall we proceed?
Is that a question for me, ma'am?
Yes, ma'am.
Yeah, we can.
Thank you. I would like to share a latest news on artificial intelligence. And this update is shared by our yesterday's speaker, Mr. Amal Rajji. The French artificial heart maker Carmat announced on Tuesday, September 21st, 2021, exactly yesterday, and that it had carried out the first implant of its ASAN artificial heart in a woman. And the company said the procedure had been performed at the UFOL Health Jewish Hospital by University of Louisville physicians in the United States. It is the first time ever that artificial implanted heart device has helped a woman suffering from heart failure. It proves the advancements in the artificial intelligence in our future perspectives. Thank you, Amal sir, for the information and interest towards the profession. Thank you so much. Very good morning to all. I welcome back to the session 2 of second day online faculty development program, Internet of Things, Revolutionary Impact on Pharmaceutical Education and Industry, 21st to 25th September 2021. It's my immense pleasure to introduce the speaker of the session, Mrs. Kavita Ayala Somayajulu, Head Pharma IT Compliance. She completed her MCA from Andhra University in 2000 and she has several industry certifications in the quality, compliance and regulatory domain. Ma'am worked as an associate consultant in Tata Consultancy Services for 21 years, 4 months. At present, Ms. Kavita ji is rendering her services as Head Pharma IT Compliance in Tata Consultancy Services, and her experience spans across consultancy and advisory in compliance areas of pharmaceutical companies. In her experience of over 2 decades, Madam has been associated with global pharmaceutical companies and working as an expert in the pharma IT regulations. Madam brings domain knowledge of the pharmaceutical business, processes and expertise in the regulations. Ma'am, yeah. We'll be waiting for just a fraction of moments.
Thank you, ma'am, for the warm words and the welcome.
Ma'am, just a minute.
Yeah.
We have stopped in between. So, Madam brings domain knowledge of pharmaceutical business processes and expertise in the regulations pertaining to these business processes like good manufacturing practices, good clinical practices, good laboratory practices, good distribution practices, etc. She has also been working with pharma companies in implementation of new age technologies like artificial intelligence, machine learning, robotic process automation, cloud adoption, Internet of Things, blockchain, etc. And the challenges thereof required for the adoption of these technologies in a regulated industry. I invite Madam Ms. Kavita Ayala Somayajulu garu to deliver her talk. Ma'am, now I hand over the session to you, please.
Yeah, thank you so much, ma'am, for the warm welcome. And good morning, everyone. If you're all comfortable, it'll be nice to see you all also, if you can switch on your cameras. And so today, the intent of this talk, I would say, is to have a mutual understanding you all bring the experience of working, or probably as either working in the pharma industry, or you are in faculty, so you bring that education knowledge. I'll try to bring my practical knowledge from the industry, and we'll try to make it a learning session for each of us. So it's not a talk. I would like to put it as a learning session where we will all share our experience and learn together.
Sure, ma'am.
Yeah. And I'll just share my screen. Yeah. So I don't have a complete background of all the various areas that you all come from, but at least based on the understanding that I got from Dr. Krishna, you are all mostly either a faculty or you've been involved in the pharmaceutical value chain somewhere. So before we jump into what are the challenges for the adoption of the artificial intelligence, I think we'll just do a little bit of a take a step back and talk about what artificial intelligence is, and then what does the pharma industry does? What does the pharma industry do? What are the various steps in the life cycle of the pharma industry? So that's something that let's start with. So I want you all to volunteer and give your view on what does the life cycle of a pharma industry look like, because you're all involved very closely with it. I want to hear your side of the story.
I request the delegates to please introduce yourself. Miss Leena, kindly introduce yourself.
Good morning, ma'am. I'm Leena. I'm doing my PhD, so I'm a research scholar in the Department of Pharmacy Practice.
Dr. Swapna, Dr. David, Dr. Vijay Lakshmi,
Yes, good morning, madam. My name is Swapna. I am a research scholar in JNTU, Anantapur. Department is pharmaceutics.
Okay. Thanks, Dr. Swapna.
Thank you, madam.
Yeah. Anybody else who would like to introduce themselves?
Good morning, ma'am. I'm Dr. Sukhwant Singh.
Sir, your voice is echoing.
Am I audible?
Yeah,
Yes.
yes, you are.
Yeah. Ma'am, I'm Dr. Sukhwant Singh. I am an associate professor at Sagar Institute of Research and Technology in Science Pharmacy at Bhopal.
Okay. Anyone else?
Hello, good morning, all of you. I am Professor AICTE from DKT Yashwantrao Sawan Polytechnic in Silvanji, basically from Maharashtra.
Okay, sir. Your good name? Sorry, I didn't catch it.
My name is Ravikant Hatigine, RA Hatigine.
Ravikant. Okay, sir.
Yes.
Yeah. Yeah.
Sukhpat Singh ji,
Yes, ma'am.
please introduce yourself to the speaker.
I just did, ma'am.
Yeah. He said he was a research associate.
Vijay Kumar sir.
Yes, madam, I am an associate professor.
Oh,
Okay.
sorry.
Good morning, ma'am, and good morning, everyone. Myself, Dr. V.K. Vansal, and I am assistant professor in pharmaceutical department of Lachu Memorial College, Jodhpur, Rajasthan.
Yeah, thanks, sir. So I think largely I see a lot of assistant professors here and some researchers. That's
Yes,
at
ma'am.
least how I would summarize, the set of delegates. Yeah. So because I see a lot of research associates here, so I want to hear, how does a drug discovery happen, or before, probably let's say even the COVID vaccine that we all have taken, before it reached into the market, what is the life cycle that it undergoes? I want to hear from you, you all, as a learning. Anybody who wants to volunteer and explain your understanding, your view?
Ma'am, you name someone. We may volunteer. We may always speak on that if you want.
Okay,
Sir,
so
you can please.
yeah,
Sir, you can take the lead. Sukhwant sir.
Okay, ma'am. Okay. Ma'am, in fact, the previous lecture was started on the same track, so I'll just take the excerpts from that only.
Okay.
That am I audible, ma'am?
Yeah,
Yes.
you are audible, sir.
Yeah. Okay, ma'am. So the process of finding out a drug starts from identification of the disease first. So first we have to identify what is the problem or what is the disease we want to target. Then we identify the protein which is responsible for causing that disease. Then we undertake CAD studies just to I'd find a suitable drug which is targeting that particular um, biomolecule or a protein for that matter.
Right.
And after that, when we undergo that studies, we identify the lead. Then we go for synthesis of the drugs. Then we go for formulation and scale up. Then we file for the investigational new drug after the animal studies. After the IND application, we go for the clinical trials related to humans. That is the phase 4. Then we go for NDA. That is the new drug application. And after new drug application, a drug is finally approved for introduction into the market. And after market, there goes the post marketing surveillance or the PMS, which is the stage 5.
Right. Yeah, that's probably what I also have here as just a summary, like you exactly mentioned. The reason why I actually wanted to go through this was to see how we distribute the value chain into multiple divisions. One is your initial one is your discovery and development. Then you have your clinical trials. Which phases, starting from the animal testing to the 3 phases of clinical trials. In between, you file the IND and the NDA. Then let's say after the regulatory approval, not necessarily FDA, any regulatory approval, you will go for a bulk manufacturing, sales and marketing, which also includes your supply chain, and finally your post-marketing surveillance. So I've tried to divide them into the various units or value chains of the value cycles. You first do the discovery, then your clinical trials, your manufacturing, sales and marketing, and surveillance. And regulatory compliance is something that almost spans starting from the clinical trials, running up to the surveillance, which wherein you do your adverse event reporting, your market intelligence, market surveillance. Everything is guided by the regulatory industry, so or by the regulator. So that's how we see it. The whole intent of trying to divide these into the various value chains is to see that today artificial intelligence is kind of trying to make impact or is impacting almost every part of this area. I mean, starting from, we have seen artificial intelligence helping in the discovery and development, enabling the clinical trials in terms of the clinical trial analytics that are run to understand the outcome of the clinical trials, enabling the patient recruitment for the clinical trials. Then getting into the manufacturing space, they are helping in just a minute. Yeah. So in the manufacturing, they are helping in quality control of the drug that's being manufactured, predictive maintenance of the manufacturing shop floor equipment. So it's kind of impacting the entire value cycle, starting from the disease identification to the point of surveillance. So it's playing a game end. That is what I wanted to start with. That is why we started this discussion on the pharma value chain. So thank you, Sukhwant-ji, for putting up a good summary on that part.
Thank you, ma'am. Thank you.
Yeah, we would like to we'll watch a small video.
Sure, ma'am. I request the participants to mute yourself and off your video mode.
Yeah, is the screen visible for all of you?
Yes, ma'am.
Yeah, and you can hear the audio also?
Yes, ma'am.
Okay.
Okay. Right, I'm going to try and replicate this here. after a great start. I tend to kind of just avoid doing sketching and writing now because it's not really worth it if you get something like that. Anything you could do that would just make my hand do what I want it to do and be able to sign my name would be an incredible thing.
How do we even just begin to help her overcome this particular symptom of her tremors and helping her be able to regain her writing ability, her drawing ability? So what I'm doing is I'm making a very rough prototype, and what this does is I can connect into it through these wires, these tiny coin cell motors. So these motors will vibrate. I personally doing is it's short circuiting whatever feedback loop there is between the brain and the hand that's causing the tremors. So the idea is if you are distracted by the vibration, are you able to write better? I don't know. It's affecting something. I don't know quite know what's happening. I'm onto something, right? I'm onto something. Jesus Christ.
It makes me forget that I have a tremor. I've actually just written my name for like the first time in ages.
I can't believe it.
Mum, it's called the Emma.
Yeah. So what did the video you just saw is about a lady who was 29 years old and she was affected with Parkinson's, and she is a designer. So being affected with Parkinson's, her hands were shivering because that's how the signals from the mind to the body go, and she couldn't draw drawings. She couldn't draw even a straight line like you saw it. But a small utility with some IoT sensors and artificial intelligence helped her, overcome that small challenge. And for her, it's a big thing. But for you and me, hands trembling might sound like a small challenge, but it actually helped her gain her life back. At the age of 29, she was not able to draw as a designer. The small project that was created with the help of the latest technology helped her, actually gain back that confidence, be able to create the drawings, which was her profession. So that's one small example of how, artificial intelligence has started impacting or helping patients in a positive way. And I say impacting in a positive way, bringing out a positive outlook to patients, saying that, okay, the technology is helping me and is enabling me to, overcome my challenges. And that's one powerful message of what AI or IoT, and in fact, all the digital technologies have been able to achieve today for the patients. Anything you want to share your feedback or your viewpoints?
Delegates, please. Ma'am, we please continue.
Yeah. So that was just to see one example of the power of AI. Now, before we jump to, more onto what is AI use cases, how they are being used in the industry, what are the challenges, I want to hear from you all. What is your understanding of artificial intelligence? How do you perceive artificial intelligence from your view? Maybe I'll take Dr. Swapna. You want to take the lead here?
Ma'am.
Okay, you can give us. Yeah, please, ma'am, go ahead.
Because I have a small experience in teaching, just 5 years only. But these are all new technologies for me.
Okay.
To learn all those things, actually, I attended this FDP, madam.
Sure, ma'am. It's encouraging to hear. Anybody else
Yes,
who
madam.
wants to, give their thought on what artificial intelligence means to you as a word? I mean, let's leave the technology behind, but the word as it is, I mean, what does it relate to you? Lena?
Ma'am, I would like to say a few words.
Yeah, please.
Ma'am, artificial intelligence is a new methodology in which we use computers and self-thinking machines, and with the help of that, we can make easy of human activities.
Yes, sir. So that's a good summary. And I mean, for example, we all use the simplest example we could talk is about is we all use our mobile phones, and when we do these text messages and sending SMSs, there is an autocorrect feature. We all see. If you type a spelling wrong, all it does is it looks into the dictionary, the closest match for that word, and in the context, and tries to replace it. So basically the machine is trying to emulate the intelligence of a human being and trying to act with the artificial intelligence. That is what artificial intelligence, if I have to sum it up in a very, very simple statement, that's the simplest example you can see of artificial intelligence. So the machine is trying to think like you, like me, and trying to replicate those actions. Now the point is, machine still being a device cannot learn on its own, so it has to be taught. So like from our younger days, we all have been taught in schools. We first were taught how to crawl, walk, and then taught ABCDs, small words, and then we learn big words. So machines also are taught with the knowledge of the humans, and step by step they kind of keep enhancing their knowledge. So for example, if I have to go back to the example we saw about this Project Emma, or the lady with Parkinson's. So the machine is when the brain signals that your hand has to tremor to the right, the IoT one will give a signal that send electrical impulses which will tremor it to the left. So a right and a left gets neutralizes, and it automatically comes in a straight line. So that is how your body is reading the signal that is coming from the brain, not the body, the device, and it is kind of giving a counter signal based on what is coming from the source and kind of neutralizing it. So that's one example of what we saw in the artificial intelligence video a while ago. And the autocorrect feature in SMSs in Microsoft Word that you use, and the simplest examples of artificial intelligence. So a machine emulating a human being, trying to use that knowledge of the human being, getting trained on it, and using it as what, in a simplest term, I would put it as artificial intelligence. I think there is a deeper dive session in the second half on artificial intelligence, where you will probably get a more insight into the technology, how it is done. But I think for now, because our focus is more to look at the challenges, I just wanted to start with a brief insight to what that means for the audience, and then we'll move into the various scenarios where artificial intelligence is being used, how those challenges are coming, what are those coming, and how we can address them.
Ma'am?
Yes, please.
Can I ask your video mode on, please?
Yeah, sure. Okay.
Thank you.
Yeah. So we'll talk about some of the use cases where we have seen artificial intelligence being used in the pharmaceutical industry. So let's start with that, and then we will go into the deep dive. So one example is the speed up of the discovery during preclinical trials. So we have a lot of research associates in this forum, in the list of delegates. So since you guys are all working on research, you would appreciate the fact that, there's a lot of effort that goes into the research to see whether, one protein can impact, one molecule that is identified, a biomolecule can impact a protein. How does it impact it? What is the dosage that should be given? And you do all these on the lab levels testing, or sometimes in the animals. So during this, a lot of effort, and when you have to do on live scenarios, you spend a lot of effort in identifying how every reaction happens. Now there are intelligent software that are coming into the market, where it can tell you that, for every cell, when you give, let's say, X percentage of the medicine, how the cell is behaving. When you change that to X plus 1%, how does that behave? A delta percentage of X, how does it behave? So it kind of tells you whether, even without going into the real life scenario, and a simulation of the software will tell you how a specific protein is being impacted by the molecule that has been fed to it, or the medicine that has been given to that, and how the cell is reacting. So the entire preclinical trials process is being enhanced. So I'm not saying it will become, you would just do that without testing on animals, but the entire simulation part is helping to speeden up the trials. So for example, one more I'll tell you, there is an artificial skin which is made available in the market today. So let's say you are working on, some medicine for a dermatology thing. So rather than trying to test it on human skin or animal skins similar to the composition of the humans, you can use that artificial skin and run your preclinical trials. So this skin will tell you how the interactions will happen, and they are kind of helping in speeding up the preclinical trial phase itself. Similarly, creating the cure for complex and rare diseases. So Alzheimer's, Parkinson's, these are very complex as well as rare diseases, and you would appreciate today, even today, we don't really have a cure for any of these. I mean, there are some medications just to stop the symptoms from worsening, but there is no cure. So there are algorithms that have been created today which can help these pharmaceutical organizations to identify the cure for these diseases. So there are, I was recently reading it. I'm not a research scholar like you guys, so I may not be able to use the correct terminology. But for Parkinson's and Alzheimer's, they're trying to find how the protein that kind of causes this can be modified in order to prevent the disease from happening. And they are doing a lot of using a lot of artificial intelligence things which use the simulation, the genome analysis of the gene, etc., to see how these diseases can be cured or avoided as well. So that's some examples of how they are being leveraged in the discovery and the development area. So since we have a lot of research scholars, have you seen such softwares or encountered such scenarios in your real life experience? I want to hear from you people.
No, ma'am.
Okay. And any of the lot of assistant professors, I'm sure you're also involved to some extent in research and in your day-to-day theory. Have you seen that? So one example is a tool called as IBM Watson. So it's a product from IBM. IBM is an IT company. Watson is the product where it has been helping in a lot of doing this genome analysis, doing this simulation in the computers, based on the various technology driven technology driven simulations basically. Similarly, the Watson also has a patient recruitment for clinical trials. So now we all understand that there's a lot of clinical trials. There are 3 levels of clinical trials that happen outside beyond the preclinical. So where you have to recruit the patients. The phase 1 is healthy patients, phase 2 is patients who are suffering, and phase 3 is a larger criteria team. patient recruitment. So what this artificial intelligence tools are doing is they're evaluating the patient profiles, and they're seeing whether this patient profile actually meets my criteria for this clinical trial. So let's say, for example, the age of the person, the sex of the person, and their current past clinical history. All of that are actually required to understand the fitment of that person for a clinical trial. So now these artificial intelligence tools, once you feed the patient information profile into the tool, it kind of helps in identifying whether this is a right fit for the clinical trial, not a right fit. And what industry parameters says, the recruitment time has been reduced by 30% with the usage of these tools. In order to, with the tools, it's able to recruit the patients faster for clinical trials. And especially this becomes more critical when you're doing a phase 3 clinical trials, because you go for a larger population. So when you're hiring around 30,000 patients across geographies, across races, these kind of tools help in speeding up the entire process. So rather than me having to spend, or a pharma company having to spend 6 months of duration just for the patient recruitment, they could do it probably in 4 months. So that's one example. And clinical trial data, you all being researchers know that a lot of clinical trial data is generated. So once this clinical trial data is generated, we run the analytics on them. We do the statistical computing of the data to determine the outcome. So basically, you're not going to run one by one patient data to determine whether this is working, right? So you run analytics, okay, on this age group with this origin. So let's say Asians. How Asians may respond to one drug, the Caucasians or the Americans will not respond, because that's what they have found over a period of time, that your race also has an impact on how the drug really works on you. So these analytical tools use the intelligence to determine, okay, how is it that an Asian race is responding? How is it that people from an age group of 12 to 18 are responding? How is it that old people are responding to a drug? So you don't have to do anything manually now. There are artificial intelligence algorithms which are based on mathematical standards. These algorithms actually run the entire analytics, and then they give you a result that, okay, this drug has been better on this age group. If you remember, the Covishield trial reports when they came, so they were run on 2 different types of trials by AstraZeneca, and we had some news in the paper that a 50% dosage was more efficient and effective than a 100% dosage of AstraZeneca for the Covishield. And there was a lot of hue and cry in the industry because AZ tried to hide those results. So how do you think they have identified these kind of results? There are AI algorithms that they run on the clinical trial data. They see how the dosaging is impacting, which dosage is a better fit, and all of these are done using the AI machines. On the manufacturing side, quality control. So you'll see, there are machines where when you're manufacturing any vaccine, again go back to the COVID vaccine, you need to maintain a proper thermostat temperature, the humidity that should be there in the processing plant, the atmospheric pressure. All these parameters do matter. So now there are actually tools at the shop floor level which keep monitoring your temperature controls, your humidity, your pressure controls, everything. You can set it. You want it in every second, every minute. So there are devices which you place on the shop floor. They keep pulling the information. So you want to monitor the temperature every 1 minute. So they will keep pulling the temperature every 1 minute. And then once you see that the temperature is going beyond limits, they will immediately raise an alarm. Or there are some tools which can automatically adjust the temperature also. So like your Pfizer vaccine, it has to be stored, I believe, at -27 degrees. Is that what it is? I don't remember exactly. no, it is at minus, something in that range. So now when this Pfizer vaccine is being transported, not just in the manufacturing shop floor, being transported, you have to make sure that the temperature limits are maintained, right? So you have these IoT and AI devices. So the IoT devices read the temperature, they push it into the AI software. The AI software, whenever it sees that, the thresholds are not met, it will send a message to the IoT device that increase the temperature, or the temperature device that, your temperature needs to be reduced further to say minus 2 more degrees or 3 more degrees. And that is how, so without even a human intervention, the entire supply chain, the entire manufacturing shop floor is controlled. So you don't really worry that, is the temperature going beyond limits, because there are controls in place which monitor the parameters and then can also modulate them to say that, okay, your temperature needs to be reduced, or your pressure is going beyond limits, you have to fix it. So those kind of tools are coming in, tools and supportive systems which do that. Now, any questions? I mean, I forgot to say, if there are any questions, you can just stop me and, we can, I can try to help address the question.
Ma'am, there is a question in the chat box.
Okay, I can't see the chat box because I'm in my screen.
I'll read it for you, ma'am. I'll read it for you.
Yeah, ma'am, please.
Can AI be used to determine results with small population of 100 to 200 patients?
Yes, it can be used to determine the results even for small population, because for an AI machine, the population doesn't matter. What matters is how you have trained the AI machine. So, for example, I'll give you one very simple example. So I actually wanted to cover it through the use cases. So when you do a search for a doctor in Google, okay, you all can do it on your respective machines. Go to images.google.com and search for a doctor, DOCTOR. And the first set of images you will see is, and this was recently rectified. Otherwise, initially you could see only all white people. So because their AI machine was trained with all, Google being, an American company, they trained their artificial intelligence program to look at all the white images, and it was fed all the American doctor images, etc. So when the minute, Google was told a doctor, the first image that it came to its mind was that, okay, all doctors are white. So it would put all the white doctor images. And this was kind of identified and notified to Google. So they changed the training set of that tool. They said they included Indian doctor pictures. They included, the African Americans, the African doctors. And if you see, even if you see, the majority still is English or white people. I wouldn't say English, white people. And then you see the other races. It's because on how you train the AI machine. So the training of the AI machine, if you should use a good data set, so 10,000 records or something of your, once you use that to train it, even if it has to identify from 10, it will use that knowledge to apply. So application of the knowledge doesn't have any limitation on the number of records. But the training of the AI machine is the most critical factor for the successful working of any AI algorithm or AI software. Does that answer the question? Hello?
Yes, ma'am. You are absolutely fine.
Yeah. Okay.
Yes, ma'am. Thank you so much.
Yeah. If there are further questions, just please let me know, because I can't see the screen. My screen is shared.
Sure,
so
ma'am.
the chat window. Yeah.
Sure, ma'am. I'll be reading.
Yeah. Thank you. Yeah. Now, do you all understand about this predictive maintenance? What does that mean? I mean, just from the terminology.
Maintaining upfrontly, or you're going to say that the machine is going to slow down maybe in the next coming week.
Right. Exactly. So maintenance is a very, very important aspect on the shop floor. I mean, starting from, the calibration of the shop floor equipment to qualification to maintenance, it's a very, very important factor. Now, there are two ways of doing it. Predictive maintenance is what we do, like your, your annual maintenance contracts. Assuming that someday it will fail, you can call somebody and ask them to check your machines. Okay, but how often should you do that? So that is where you will have these IoT devices on the manufacturing shop floor, which will keep on reading, the parameters and say, let's say you're talking about a chromatography system or maybe some weighing scale on the shop floor, etc. So the precision is very, very important. So it will keep on reading the current values at it, and whenever it sees that the threshold values are not met, or it is the last maintenance was done 30 days back, it is due for a maintenance. That is one way. And the other way is the temperature is not holding, or the chromatography system is not giving the values within correctly the limits. So it will keep on looking at the data, and then it will give an alert that okay, either the timeline has passed, or you see these are the various parameters which are not showing correctly. So there is a requirement for a predictive maintenance. So you predict the maintenance before the system actually stops. So rather than waiting for the system to break down and then going for a maintenance, you are actually going too upfront, too early. So if the system might break down in 50 days, you don't want to do a maintenance every 30 days because that's not cost effective. So you might want to look at the threshold when the system gets due for a maintenance and do the maintenance. Similarly, there are other examples also where you define the golden batch, the batch when you're doing the manufacturing in the drug manufacturing, right? You first define the golden batch, which says what is the recipe, and based on the recipe you define the golden batch. Now that golden batch creation also, there are multiple ways of doing the golden batch manufacturing, doing manually, changing the parameters and building the batch, manufacturing the batch, doing the quality control and the quality test, and then saying okay, this is the perfect batch. The other is building using your AI tools, which can keep on varying the various measurements from the recipe and say which is the perfect batch that is being manufactured, because it's all discrete, right? So you have to give the quantities, the ratios in which it is done. So these also help the looking at the purity of the drugs. All of that also today is being done by the AI machines rather than humans going ahead and doing the testing. Yeah, there will be a human in the loop in terms of ensuring that the results that are coming are correct, because you still have to somebody has to say that this machine is working correctly. So there is a human in the loop, but the tools are doing these kind of activities like maintaining the doing a predictive maintenance, defining the golden batch parameters, or if you're seeing that based on these parameters the production has gone down. Let's say the efficiency of the production unit is coming only to 80%. So now what parameters should be changed so that the production unit maintenance efficiency can go to 90%. So they are tools. So one example of this is sorry, I just forgot the name. I'll just let you know.
Ma'am, Dr. Sukhwant wants to talk to you.
Yes, ma'am. Yes, sir, please.
Sukhwant kindly. I think there is a technical issue from his side. He posted in the chat box. Can I read, ma'am?
Yeah, yes, please, ma'am.
Is that two example of AI and machine learning?
Sorry?
Is the tool, is that two example of AI and machine learning?
Yes.
He was talking about, please take up the example of Professor Hawking and his artificial voice, if possible. He has two suggestions. One question and one suggestion.
Right. So yeah, if you see Stephen Hawking's case also, he was, whatever speech he was talking, right, the machine was trying to understand that and printing it back on the screen, right? So even that is an example of artificial intelligence. The tools that Dr. Stephen Hawking used to speak to the computer, because if you hear any of Dr. Stephen Hawking's recordings, for us to decipher exactly, it was very, very difficult. I mean, because the voice is not very clear. So the tools were built which were using Dr. Stephen Hawking's voice to record, to also type. All of these are also part of the machine artificial intelligence techniques. Does that answer the question, Sukhvantji? I know you have technical challenges, but if you could either answer on the chat or whenever your audio is back.
Yes, ma'am. Please.
Yeah. Right. And then
Yes, he answered, ma'am.
Okay. So he is fine?
Yes, ma'am.
Okay.
He's satisfied.
Okay. Thank you. And then looking at the sales and marketing area, sales and marketing has, I mean, this is something that people have been doing forecasting for quite some time, but now they are doing more precision driven forecasting as to, in India, like diabetics is the highest population that is anticipated to grow. I mean, at least the pharma companies, their research states that India is going to be the probably the capital of diabetics in the next 20 years. So they have done that analysis based on our lifestyle habits, our food habits, our geographical distribution, also the nutrition levels that we have. Probably one of the indicators for them also is that we are the highest sugar consumption country in the world also, both producing and consumption as well. Not that only sugar leads to diabetes, but if your consumption is high because of multiple parameters. So they are doing this kind of market analysis to target the population, saying, okay, that this is my next market. So what area should I target? Even here, the urban population is their target rather than the rural population. For multiple reasons. The rural population's lifestyle is not sedentary. They still do go to the fields, do a lot of work. But the urban population, and then access to healthcare is another reason for this distinction between rural and urban. Urban, you have access to healthcare, you have the means to get tested, both the financial means as well as the infrastructure. So you will get identified early, and then if you see the markers for also have been changing. If you look at the Diabetes Association, American Diabetes Association, they have reduced the markers. I remember from 140, now they're at 100. So the metric levels have also been changed. So these are some of the ways in which they are trying to look at the market, do a market analysis, do their sales forecasting, and see also based on the forecasting, how is my performance happening? Are people really buying my product equally? So that is one part. And similarly, the supply chain optimization is also linked to the same story, that if I don't want my drug to be sitting in a pharmacy store without being sold, right? Because that doesn't give me. So I need to understand where my market is and make sure my supply chain is routed into that direction. So, or into that area. So that is how also they are looking at the supply chain optimization using the artificial intelligence. So they, if you will all see, whenever you get a blood test done, or even you go to a pharmacy, the minute then you kind of keep getting ads, especially if you look at Google, you will get ads that this blood test is, you open Google and do let's say you want to get a blood test done in Thyrocare. The next minute there are so many blood test centers that are available, they keep showing up on your screen. So they actually use your knowledge that you have put in. Or if all of you would have also seen, you go to Google search for one item today, that item keeps popping up in your screen every time, even after probably 6 months down the line, because somebody is capturing that knowledge and running artificial intelligence on that and saying, okay, hey, this person was interested in buying X thing. So let's show him, similar things on that area so that we motivate him to buy. So that is how they kind of do. One more thing, I mean, though it's not really relevant to the pharma industry, but I just wanted to give you an general industry example. Most of you, if you try to book train tickets, you go to Make My Trip and try to look for a ticket. You enter a source, you enter a destination, you enter the dates, etc. Now you find some price. You go to the mobile. Now if you did that from your laptop, you try to do it from a mobile now. So what, they have softwares which can do that intelligence, that your laptop and your mobile are coming from the same IP address. So it is the same person, and they can actually trace you. That it's the same person who has actually tried to look at the price from the laptop. Now he's trying to look at from the mobile, and you try to use a third device from your home network, it will tell you, because they have that intelligence which looks at their IP devices, the source and destination, the same dates that you have entered, the number of people. So they kind of zero in on that this is the same person, and kind of, try to reach you out saying that this is the price that is available, you can buy it. So they actually keep monitoring whatever you're doing. In fact, and probably, from the time we wake up till the time we go to sleep, all of our activities are monitored. That's a different story, not in context of this, but that's how artificial intelligence is being used everywhere. Okay. Any questions?
Ma'am, we have a question from
Yes.
Soumya.
Yeah,
Ma'am, can we use AI in technology transfer and scale up by any means?
When you say in technology transfer, what does that mean? I mean, I didn't get the question. Is it more to, when you say technology transfer in terms of transferring the technology of, let's say, a vaccine being built from one company to another? Is that what you're saying?
Soumya, ma'am?
Yes, ma'am. In scaling up the formulation from laboratory scale to commercial scale, like that, ma'am.
Yeah, so their AI is more used. You can use it. It's more used in terms of, like I was explaining you, right? When you're doing a formulation in a lab level, it's a very small formulation. But when you want to do a large scaling, that is where I was talking about the golden batch example. So any pharma company, when they do a large scale formulation, they first create the golden batch and they look at the yield for that, and also, make sure they want to also optimize the yield. So this is where the AI plays a large role in helping with defining the golden batch, in the yield optimization, because when you're doing small scale, yield optimization doesn't play a big role. But like looking at, the way the vaccines is one small example, the way vaccines have been being manufactured, your yield optimization is very important. If your yield is only 80%, 20% is your wastage. So this is where it helps in scaling up by optimizing the overall recipe, the overall yield that is coming from the manufacturing. So there are tools which are there already, and there are specific companies which do these kind of AI things.
Ma'am, I would like to interject once again,
Thank you.
if you permit.
Yeah, yes, sir, please.
Sorry, ma'am, for the previous audio disconnection. I have logged in from my mobile as well. Ma'am, I have a question regarding the surveillance type. Like, for example, we all are now these days using health bands in general,
Yes.
just to keep the monitoring of our, various things like monitoring our oxygen, monitoring our heartbeat, etc. Now, ma'am, my question is that comes to my mind is that is it possible that this data will be used up by various, insurance provider companies, where they'll be keeping a track of how my health is in general, and whenever they offer me, whenever I go for a policy for securing my health, they may take up those data and offer me a different premium for that particular health policy. And
Yeah.
is it ethical to use this data, ma'am? Kindly shed a light on this.
Yeah. So in India, at least those kind of data is not being gathered today. So, but yes, the world is going towards that direction. Before I answer your question, I want to give a small example of what is happening in the United States. So in the United States, see, you talked about health insurance. I'm talking about the vehicle insurance. In the United States, all the insurance companies, there are insurance companies, what they do is they install a small device on your car. Okay, so which keeps looking at how you are driving. So it monitors your speed, whether you're overtaking or are you not following the rules, etc. So now this device keeps on polling your data, and it goes onto the cloud and stores. And the insurance companies actually use this data to say, okay, you have been driving very well in the last 1 year, so your premium is reduced because you are a less risky customer for me. So they have been doing this kind of data already in the US, especially in the insurance section for the vehicle insurance part. Now coming to the health insurance, what is ethical? So there is a consent that is mandated. So in India, we don't have really so much stringent laws with respect to data privacy, like the GDPR in the European Union and the HIPAA in the US. So we are still, being a third world country, still not there yet. But in US and Europe, any data that is collected requires a data privacy. Otherwise, there is huge legal obligation to it. There are penalties that could be levied onto the companies who are doing. So any data that is collected has to be permitted, or you have to give a consent form that yes, this data you are collecting, and they also have to state what is it that they're going to use it for and whom they're going to share it with. So that is the minimum consent that is required, at least in the European Union and the United States for sure. In fact, Japan and China also, the data privacy laws are a little bit stringent when compared to India. We are still not there. So without consent, no data has to be gathered. Okay, that's one question. Now, they are getting there, not yet, but they will get there where, so today in India, your health insurance premium is dependent on your health profile. So dependent on your, if you go and ask for a health insurance premium from any vendor, they'll ask you to do a health check. They look at your health parameters, and then depending on that, the insurance premium varies. So that is already there, but it is not being done proactively right now. It is all a reactive. You go for an insurance provider, they do the parameter check, and based on that, they offer you an insurance coverage and a premium amount. But we are getting there slowly. So there is this concept of EHR and EMR, electronic health record and electronic medical records. India is not there yet, but there is a lot of emphasis happening in the developed countries already on this. So for example, in India, you go to a doctor, the doctor gives a prescription, you keep it with yourself. And then next time you go to the doctor, if you don't carry the prescription, the doctor has no idea of what happened to you. And they have no recovery knowledge of that. Or if you go to another doctor, he will not even wonder to ask you what happened to you last time. That is how it works. I mean, that is the mode of operations, not criticizing, but that is how we operate in India. But in the US, they're trying to link all the electronic health records of a patient in such a way, in a uniform manner. So even if I go to X hospital today, a Y hospital tomorrow, or a Z hospital the day after, they will have the entire record so that they can use that to treat me correctly. And there are also AI mechanisms. So let's say, I enter into an emergency hall today, suddenly, and the doctor is there, but they will have AI machines which will look at the entire health history of mine, the EHR, EMR records, and say, okay, this lady had maybe some problem and she went to a doctor, heart problem, got a heart test done, let's say an ECG done. There was a problem with the ECG. Then she went and saw a cardiologist. This is the medication she is on. This is her BP. So all that records is there. So based on that, the AI machine or the doctors also can predict what could be the possibility. So they are talking about, so although it's a big initiative and very expensive, because you need to agree on a standard in which you will gather all the information, the electronic health records. And these are all available in a common database, which should be available across hospitals. So in fact, in US, if you go to a doctor, the doctor will have the entire history with them, and they have to retain it. I think, I don't know, but at least somewhere in the span of 7 to 21 years, every patient record, they have to retain it for that duration. So they don't actually give you your health records. They only give you a copy. The original lies at the doctor's office, however small a doctor may be, because that's the rule in that country. So here also, we are not getting there, but the electronic health record and electronic medical records concept is picking up, where they are trying to keep all of this together. And there will also be an integration with these vehicles also. Let's say an accident happens, so the world that we are looking forward to is let's say an accident happens, immediately your mobile, the AI machine, based on the crash sound that happened or something, it will pull into your EHR, EMR. It will immediately notify the doctor, the nearest ambulance service center, and so that you can be called in. That's where the AI is going today. There are things going on. I mean, for India, maybe another decade is what we are looking at. But some of these things are already implemented in the Western world. Does that answer the question?
Sukhwan, sir?
Yes, ma'am. Thank you very much. Maybe in India, the data, maybe because it is India, maybe because we have very large data sets, maybe. That is one of the reasons why we still have not picked it up.
No, we don't have data sets. That
Yeah,
is the
because
problem,
ma'am, the
actually.
population of India is a huge population as compared to the Western world.
Population is huge, sir, but we don't have data sets because for us, all is unstructured and ungathered data. If I ask you, will any doctor have your medical record? I can give a guaranteed no as an answer.
Yes,
Now,
ma'am, sure.
slowly, if you go to a hospital, they have started keeping a record of your prescription, etc., for future. But that's also very at a naive stage. So unless we have all this data collected together, I mean, for machine to really evaluate, the data is the primary part for it. And in India, right now, the status is we all have unstructured data for all of us, and unless we bring it into a structured in any format, into one common system where it can all be polled, running these kind of AI machines will be very difficult.
Thank you so much, ma'am.
Yeah. And then coming into the surveillance part, looking at pharmacovigilance,
We have a question from Leela Mathuri.
Yeah, sure, ma'am.
Ma'am, chatbots comes under AI?
Yes, they come under AI. So there are two kinds of chatbots. One, which only have a set of question and answers, and they'll only answer that to you. Those chatbots are more of robotic process automation. So something that is being done in the same fashion is being automated. But there are chatbots which are intelligent. When they are intelligent, I mean, let's say, they will read from the literature and they'll try to answer a question. And they'll ask you if this answer meets your question, response to your question or not. If not, they'll go through the literature back. So they are trained, and then they keep on enhancing themselves based on the new literature that keeps getting added. So those fall under the AI category. The ones that are pre-trained or which have a pre-literature that they look at it and answer, those fall under the robotic process automation. So that's not considered as artificial intelligence. It is a repetitive task being done. So you give it a set of questions and it has a set of answers which it will pull from and give it to you. But there are some, like I said, which do a self-learning. Those are actual AI driven bots. Does it answer the question, Lina?
Yes, ma'am. Okay, ma'am. Thanks. She replied in the chat box.
Yeah. So now the pharmacovigilance. Pharmacovigilance is the phase 4 clinical trials where you are actually putting up a vigilance of the drug usage on the market for an adverse event reports that are happening. So we also have heard, right, during COVID that there were some adverse event reports that were reported. So the vigilance of the actual world is what pharmacovigilance is. So pharmacovigilance, one is the actual world vigilance. So what happens is, let's say, I have a drug reaction. I can report it to the regulatory agency or I can report it to the pharma company, which then does the case triaging. They kind of, try to understand the circumstances in which the medicine was taken, what are the reactions that happen. They do a medical review and then they say, okay, it happened because of whatever XYZ reasons. That's one small part. Now, but the pharma companies also have to do some good deep dive analytics and understanding of these adverse events to say, are there repeated adverse events and are we seeing some pattern? So if we looked at Covishield, there was one of the adverse event they said was blood clots. And they saw it happening. And Israel reported that, the Pfizer's vaccine people were taking, they was seeing some heart enlargement in some of the young people, some in the range of 30 to 50. So these are actually side effects. But these are not one-off side effects that, you notice and you can leave it. You have to make some correlation of those side effects and say, okay, these side effects have happened in these many populations. So these are very frequently seen. So this is where the AI is also used. And additional place where we have seen a lot of AI being used is these social media. In the social media, somebody will put up a post saying that I took a Covishield vaccine today and I had this adverse event. So what this person is doing is, this person is not officially communicating to the regulatory body or to the pharmaceutical company that he or she has had an adverse event, but he is just posting it on a social media. Now what the regulators are expecting is, you guys monitor the social media and make sure that you also report those adverse events. So now monitoring the social media is not a small joke, right? You have Twitter, you have Facebook, you have LinkedIn, you have so many other channels. So there are people are building artificial intelligence tools which are looking for any drug name and associated reaction name. So now reaction could be, I can pull up from the the dictionary, the data dictionary, multiple ways. I could say I had cough, or for cough somebody could say something else. Said I had a respiratory illness, or there are multiple names also. So these these AI machines, they have to go into the internet, search for any medicine or a reaction, relate those sentences, pull out that information, now articulate it into whether it's an adverse event, and then post it into the adverse event database. So this is something that our organization has also done, monitoring the social media, get the data from there, and then post it as an adverse event. So this is what are also being done in order to monitor the overall adverse events in the industry. So not just a personal reporting, but if I just post it on some XYZ media, the pharma companies are being liable to monitor that and respond to that also. So that is where they are also using artificial intelligence. Now somebody cannot sit and browse the websites, right? So that is where they're using these intelligent machines to do that kind of vigilance in the market. Similarly, drug adherence monitoring and the dosage monitoring, this is also being done both for the clinical trials also. See, when your clinical trials are done, whether the patient is adhering to the dosage or not is something that they are monitoring with AI based on some sensors that will look at the results. And also especially for the post-market phase, you see a lot of adherence being monitored for primarily the diabetics, whether they're taking insulin or time or not, and also the psychiatric patients, because what the industry has seen is that antipsychotic drugs are the most ones which are used very haphazardly. A patient will use it for a week, then they stop it on their own. Because of this, there'll be a relapse, then they'll come back, they'll again take the medication. So these are two areas where they're trying to do a lot of drug adherence monitoring. And if they see that the drug has not been taken, they'll inform the relative or somebody that this person has not taken a drug. An SMS goes. So they're trying to bring that control in the timely drug intake through the drug monitoring, through to these tools which are being put in. So there are small monitors which will monitor it and then communicate to the patient or their nearby ones that you have missed your dose this time and please take it. So that kind of monitoring is being done. And disease identification, we saw this recently, right? During the COVID, Israel created an app which could monitor whether you had COVID or not based on your voice. So it will ask you to speak up, and based on the voice modulation, it could identify whether you have had potential of having a COVID is there or not. When I say potential, you are either COVID infected or not. And they found almost a good 90% truth, which is more than RT-PCR. RT-PCR efficacy is only 60%. They found a 90% efficacy with that Israeli app that was created. And even there are now tools which are looking at patterns of ECGs to say, whether that person could have a potential heart attack, looking at, reading the radiology reports and communicating whether the person has had pneumonia, especially, a lung X-ray. So there are softwares which are doing this, and these have been built. In fact, India also, some young kids from IIT have built these kind of softwares in the recent past. So that's how they are trying to integrate intelligence into these tools. Even there is this one guy, one small kid in the COVID, during the COVID duration. His mom is a doctor and she was wearing those PPE kits all through the day. And so with PPE kit, we all read, right, that they don't get air inside and they are not able to breathe properly. They feel suffocated. So because his mom was a COVID warrior and she was wearing the PPE kit, being a doctor, this kid, he invented a small machine which, when installed inside that PPE kit, it releases air, fresh, cool air every some seconds, 10, 15, whatever seconds, I don't remember. And he collaborated with IIT Mumbai to do this. And so that if you wear that PPE kit, you don't get suffocated. There is air that is being pumped in. And because the device is fitted inside the PPE kit, and there is no way, that the contaminated air will go inside. He has created that device and fitted inside the PPE kit, and it was very inexpensive also, not very, very expensive. I think some 300 rupees or something is what it finally came down to. It started at 15,000, but they did research along with IIT Mumbai guys, and it came to some 300 rupees to install that device, which is very, very cheap compared to what the inconvenience the doctors are going, right? So those kind of things is what AI is helping do in the pharma industry today. Any questions?
Ma'am, there are no questions in the chat box, ma'am.
Okay. Yeah. So similarly, epidemic prevention also, there is one use case. I'll just talk about it in the next slide. So these are some of the real life examples that I have picked up from the real pharma. So ABOT has a artificial intelligence powered platform for imaging. And what this is doing is, when a cardiologist does a stent implant, right, a coronary stent implant, it gives you multiple images so that the cardiologist will know how much of a calcium deposit is there and how much should they inflate. So they are not doing any guesswork. They are actually looking at the images while doing the implant, and they can make a better precision job. So ABOT has launched this platform for imaging. Similarly, Bayer and Merck, they have created a platform which does this. Reads the images of the X-ray images, and it helps the radiologist to identify the patients with the chronic pulmonary hypertension. So because of lung infections, some patients have hypertension. So they have created a platform which will help in identifying that. So it looks at the images and identifies the patients who have a pulmonary hypertension. And then Novartis is using it for clinical trial data. We already talked about in the previous slide. Boehringer is using it in the drug discovery phase. So like we said, preclinical trials, speeding up the clinical trial recruitment. So that is what Boehringer, which is a German pharmaceutical, is doing. And GlaxoSmithKline, which is GSK, this is what they are doing it for their allergy cold flu. So most of they have a big chunk of their segment which is into allergy and cold medicines. So what they are doing is they are using AI to predict the season based on the weather changes, temperature changes, atmospheric variations, etc. They are predicting as to when the weather will be bad, so that they can upfront send a notification to their patients that you have to take a cold or a flu anti-allergen medicine so that you don't get impacted. So that is how they are. So there are two ways. One, it is patient centricity. The other is they can boost their sales and don't know how to divert their supply chain to which part of the world and when. So that is how their GSK is using. So this is almost like predicting a health situation, right? So that is what they're using AI for. So these are some real life examples from the actual pharma world where they have implemented the AI. Any questions?
Yes, ma'am.
Yeah, please.
We have a question from G. Swapna, madam. Ma'am, is there any app to know whether the patient takes medicine or not? How we know through AI?
Yeah. So there are small devices which can be implanted. So for insulin, there is a monitoring device that comes, I think, from Novo Nordisk. So you can implant it and it will tell you that your insulin levels have come down. So now it is your time for your next dose. And it can also tell you how much dosage you have to take. So especially see for patients with type 2 diabetes, it is much better situation. But for people with type 1 diabetes, the fluctuations are very high, and they will have diabetes for a long time. So managing the diabetes very efficiently is important. So there are implants which are inserted inside the skin, and these keep on reading your insulin parameters, and then they'll tell you your blood sugar levels, your insulin levels. They'll keep reading, and then they'll give you an alert. You will see an alert which says that so this data is sent from that device. Either sometimes they store it on the device, and sometimes they pull it onto a software in the cloud, so where all the data gets collected. And then there will be some software which will be running on that data and saying, okay, in the last 6 hours your insulin, your sugar levels have been stable, or this is your sugar level, so this is the amount of insulin that you have to get. So you get a message or an alert saying that this is the amount of insulin you have to take. Or if you miss your dose, they'll alert you that you missed your dose, that's why your sugar levels have gone up, your insulin levels have come down. So that's a real life example of how AI has been used for insulin, especially the diabetics.
Ma'am, there is one more question.
Mm-hmm.
Dr. Vijay Kumar Bansal, sir. Is there any AI related to teaching and students' activities?
Yes, there are tools that are being built today now. So in fact, before I come to the teaching part, there are AI tools that are being given this pandemic. We have all shifted from offline to online. So students are attending school from home. Your colleagues, we are working from home. You have been teaching from home. So now people have built AI tools which can keep monitoring your activities. For the students, when you switch on the camera, it will monitor your movements and say whether you have been looking at some different direction for a longer time. So are you cheating? It will read your facial expressions, and it will try to find out whether you're cheating or you are not cheating. They have actually built those softwares because you never know when we will return to the real world of the physical exams, etc. Right? So they are actually building those kind of tools. So now coming to the teaching world, if you see a lot of these online tutorials, right? Byju's and I don't remember the others, Classmate. So all of these also are building in AI to assess the knowledge of the student. So they don't really have teachers who will, look all these answers and correct them. So what they do is when they give you that software, right? They'll give you questions. When you answer and respond to it, now the answers are not corrected by the teachers. They are corrected by AI tools, which will look at these answers and try to gauge the areas where the child's performance has to be improved, etc. So that is where this AI is also enabling the teaching profession, and also in gauging the weak areas of the student also, because the teacher, Byju's, if they sell 500 or 5 lakh copies of their software, they are not going to hire 5 lakh teachers to correct the students or fix it, right? So that is where they invest in all these AI tools, which also correct your papers, monitor your performance, give you gaps, and give a report of your performance also, identify the weak areas. Yes, they are using those things.
Yes, ma'am. We have one more question from Harini Upada, ma'am.
Yeah.
Is the AI data monitored by a person continuously or periodically?
It is driven by the need. So see, if you have to be talked about the students' performance, right? Students' performance, you don't have to monitor it daily. You will probably monitor it weekly or maybe fortnightly. But the temperature controls on a shop floor, or your predictive maintenance controls on the shop floor, that's something you will monitor probably even more frequently. You will monitor it you'll pull the data every 10 seconds. You will monitor the performance every daily, or every 8 hourly when the shift change happens. Because when the new shift people come, they need to know what is the performance of the previous shift, right? So it's all configured depending on the need. It is not very standard, but depending on what is the need, that is how it is driven.
Yes, ma'am. Thank you.
Yeah. I have few questions for you people. Anybody can answer. The intelligence that we display, what is it called? Constance, ability, natural intelligence, or cognition? I mean, I don't have any chocolate.
Cognition.
Natural intelligence, ma'am.
Yeah, natural intelligence, right. Cognition is learning. Natural intelligence is what we display. Great. Claps to the person who answered it. The next one. Software designed to facilitate a conversation between a computer and a human end user.
Chatbot.
Chatbot. Great. Wow. The next one. Which of these is not a smart personal assistant? Siri, Cortana, Alexa, or
Cortana.
Maybe Alexa.
Any other takes?
Siri, ma'am, Siri.
Siri.
No, it is the last one. See, Siri is the smart personal assistant from Apple. If you see Apple iPhones, all Apple iPhones have Siri installed. So you can ask it a question and it will try to answer it. Alexa is from Google. Cortana is from Microsoft. These are all three smart personal assistants. Librettos is the only one which is not a personal assistant. It's an artificial chess game. It's an artificial intelligence machine which plays a chess game, and it has been able to defeat all not chess, poker game. Sorry, it has been able to defeat the best poker players in the world. So since you guys might have your phones, right? People, anybody who has an iPhone. Sorry?
कर दिया यार, ठीक है, लगे। और फोर्थ सेम का मिला है क्या आपको? अंजुलता मैम को मिला है, ठीक है।
Sir, you're not on mute. Can you
the participants to kindly mute yourself? Dr. Vijay Kumar. Sorry, ma'am. Please.
Yeah. So Siri is on Apple iPhones. Cortana is there on Windows phones, and Alexa is the smartphone from Google, smart assistant from Google, and Librettos is the only one which is not a personal assistant. It's a gaming software which plays poker, and it has won against all. I mean, it seems it had a competition with 4 best poker players, and it kind of beat all of them. And what some interesting facts about Librettos is, it would look at the poker game of that person throughout the day, right? It would play with that person the poker. In the night, it would look back into all the moves that were made by the other person and start learning from it, trying to evaluate that person's mindset, etc., and then implement that the next day. So it is a self-learning AI poker game software. Yeah. So now we'll try to come to the actual challenges that people are facing for the adoption of AI. We have talked about so many AI use cases, how it is helping. Having said that, we still have we have not seen a big everybody trying to use AI everywhere. There are use cases which are happening, people are trying to use it, but there are still some challenges that companies are facing in adoption. Now, what are those challenges is what we will try to understand here. One is the technology challenges, the first and foremost. So now AI technology means you need a new set of infrastructure, you need a new set of applications in order to deploy any AI software. So you have to make a new set of investment. All your existing softwares will not work. So for example, if I want to, let's say, improve my production yield, so I have to build this platform. I have to make sure that this platform is integrated with my existing data. It can read the data from that and then run. So my existing platforms will not be able to adjust to an AI platform. So they will stay as it is. I have to make additional investments, bring the new technology to install to bring that new applications. So we have both the infrastructure and the software challenges. So it's an additional cost to the pharma companies. Then skill set challenges. So AI being an emerging technology, you don't really find people who can build the knowledge of a human into a computer. So the simplest example is, we all work in an area, right? So you are all somebody who is a research assistant. I take care of regulatory compliance for pharma. There are people who teach people. Now, for us, we all have our knowledge in our brain, but that knowledge has to be translated into a form that the machine can understand. So we have a big gap. I have the knowledge, I don't have technology. The guy who understands technology doesn't have the business knowledge. So that marrying of, the what is there in the mind of the business or the subject matter expert, translating that into, a machine understandable rules and regulations is also a part of the skill set challenge. And I'll tell you, I am genuinely facing that challenge because I kind of take care of the pharmaceutical compliance for FDA, the FDA, EMA, etc. So there are a lot of compliance things which we do manually. I want to use AI out there. I know how to do it, but how to translate that knowledge of mine into an AI machine is where I'm challenged, because I explain this problem statement, but the gathering of the knowledge from the technical guy and his translation into the tool is a challenge. I mean, unless you do a perfect communication here, I might ask for an elephant, the guy will give me a dog. And that's what happens with most softwares if you don't translate your requirements correctly. So that's also part of the skill set challenges. The business cannot really relate to how explain that problem to an IT guy. Then you have the regulatory challenges. Now, pharmaceuticals being a very heavily regulated industry, the regulators ask for a traceability for any and every decision that is made. So if an AI machine makes a decision today and makes some changes and says that this is how it is done, whom do you put the accountability on? The regulators want accountability, they want traceability, and they want to make sure that the decisions made are correct. So now for an AI machine, at the end of the day, it's still a machine working on some set of data that it has been given and some set of knowledge it has been imparted. So for that machine to be able to make decisions is easier because it doesn't have to think like you and me. But if I have to justify the decisions made by a machine to a regulator, it's very difficult. I mean, and that is also one of the challenges why you are not seeing big time AI. So even if people are putting AI solutions in place, they put a human in the loop somewhere for the human to say that this AI solution that has been given or decision is correct. So that you can say, okay, the machine did not make the decision, the person made the decision based on this knowledge. So that is the regulatory challenges that people are facing. You have to prove to the regulators that this is correct. Then there are trust issues. When I say trust issues, the trust issues we are talking about here is the correctness of data. So how am I training my data? Is my data accurate based on which my machine is being trained? So I was explaining about the doctors, right? So it also relates to data bias. So if I give it all white doctors images, it'll understand that okay, all doctors are white, and it'll only show you those images. So I have to show some. I have to have a correct data set when I am training my AI machine. So even for clinical trial data, right? So if I am running my clinical trial data, so I have to train my data set on huge amount of data so that it can make the predictions correctly. It can learn correctly, and then it is not missing out something. So see, you and I, we have learned over a period of 20 years. I mean, from the time we started our ABC to the time you all graduated, it's at least a minimum 15 to 17 years journey. So all the knowledge that you have tried to gain over the 17-year knowledge, obviously, if you're doing a research today, you couldn't have done this research before without your M.Pharm or without your B.Pharm or without your, biology stream in your 11th, 12th or your 10th. So obviously there is a relationship, and all the knowledge that you have gathered over the years is what you are using today. Now, if you have to teach all that 15-year knowledge to a machine, it's really difficult. So your training data is very, very important aspect when it comes to training any machine. And also the algorithm. So how you have to make sure that, the algorithms that are used are transparent. So the business, you can't hide things behind and say that this is how I'll use AI. There has to be a lot of transparency to bring the trust on the AI application that is being used. Because unless the trust factor is there, you can't, nobody would want to entrust themselves to a machine, right? So that is where the trust issues come. Then the data quality challenges. We talked about EHR, EMR a while ago, right? Electronic health record, electronic medical record. So now, unless I have proper data. So first is my data set should be very exhaustive. I should have correct amount of data, or I should have all data. So the type of data, the correctness of data is one part of it. The format in which I manage. So now I put my data in a Word document. You put your data in an Excel format. A third person puts his data in an PDF format. Now, how do you train the machine? Or even if I want to, so if I want to change my, share my electronic health record from Apollo to, let's say, X hospital. Now, unless they have compatibility, they have standards. See, in the research world, you have the standards, right? You have the CDISC standards, you have the WHO naming convention. The whole intent of these standards is to bring that neutrality between people. So even if you're a researcher in India or a researcher in US, the medical terminology will be the ICD terminology. Your reporting standards will be the CDISC standards. Your WHO standards is what you will use. So that way you have uniformity. So unless you bring that uniformity in the entire data set, or at least a standardization, you will end up with data which you cannot use. Because I will write just not the standardization, right? If you go to the European world, they write everything in their local language. You go to France, you have French. You go to Spain, you have Spanish. You go to Italy, you have Italian. In India, some people, still doctors write in English, but there are situations where you have to write it in Telugu, you have to write it in Hindi or any other local language. How do you process all this data? Multilingual data. So that is additional challenge. Right. Just a second, sir.
Ma'am, if you need any 5 minutes break, you can please. I request all the participants to kindly wait for ma'am.
Give me like a 2 minutes. I'll just.
Yes, ma'am. Dear participants, all the delegates are requested to join the session with the microphone muted and video off for the third session. Attendance of 80% minimum is to be maintained throughout the 5-day sessions. Minimum score of 60% marks in the test conducted at the end of the program will only generate your certificate. Feedback link will be given to you at the end of the third session. Kindly don't miss your feedback link. At the end of the everyday sessions, we request you to be present for the photograph with the speaker in video mode. I hand over the session for 1 minute. Yes. Ma'am is back. Ma'am.
Ma'am, I'm back.
Yeah.
Yeah. Right. So we talked about the data quality challenges, and then data security and privacy. This relates, if you remember, there was a question, can people collect data? How is data privacy challenges existing? Yes. So data security, because see, most of this AI data, if you will see, they will all go onto the cloud, because there will be humongous amount of data. You can't keep it on premise. So if you are storing all these humongous data on the cloud, there could be cloud data security breaches happening. Then data privacy breaches also keep happening. If you keep looking at the search for data privacy breaches, you will see recently, I don't remember which company, I think Amazon itself had a data breach on a lot of their customer information, credit card information. Similarly, your health information is also data privacy. It falls under the personal PHI, personal health information, which is driven by the data privacy regulations in the US and the UK. So under that, if there is a data that is kind of compromised, there are huge implications of that. So that is also one challenge why people are also a bit wary about implementing AI, especially with privacy related data. Then data bias is also what I said. You want if you don't have a good data collected for the training the device, for training the application, you may end up with a biased. And see, AI applications at the end of the day are not humans. See, even humans tend to make mistakes, but the assumption is that if a human makes a decision based on all the information that they have, they will make the correct decision at that point in time, or based on the information they have at that point in time, they will make a decision. Now, when an AI machine makes a decision or gives, there is never a guarantee that it will be 100%. So, if you ask us in the IT world, AI, if it can give you an 80 to 85% accurate results, when compared to a human, it is considered to be successful. So the 15% is still a risk factor. So now achieving something more than 90 is like awesome in an AI machine, the accuracy, because it depends on how much of a data you have given it, how many scenarios you have given the machine to train it. So you have to give it multiple scenarios, multiple options, multiple codifications, language, the terminologies. Only when you give all this information and train it, that is when you can avoid the data bias and trust issues for an AI algorithm. And last but not the least is change management. We are all averse to change. The minute you talk about a change, all of us are afraid, because we are either worried. See, if I want to bring about an AI machine which to do an activity which I was doing for the last 20 years, so I automatically become insecure. I don't want somebody. I'm worried that I will be replaced. I could be worried that my job is lost, or I have to learn something. And with my so much of I cannot learn new things now because I'm old. So bringing about a change is the toughest thing. Probably the other challenges are easy to handle, but this is more of an interpersonal challenge. You have to convince the teams as to why this is needed. If you go back to the early 95, 2000 time frame, most of you would have been kids probably at that time. But when IT came, and the Indian Railways, they wanted to adopt machines, all offices wanted to adopt computers, there was hue and cry, because everybody was worried that I will lose my job. I don't know how to operate a computer. I can't use a computer. How will I learn new technology now? I am already 50. It was hue and cry. But despite 2 decades of computers coming in, we have not really seen any job losses. People slowly adapted to it. But the minute people said computers will be introduced, there was hue and cry. So you see the same situation here also. The management says, let's bring in an AI system. Everybody shuts off. No. This is all fine. I don't know how the system will work tomorrow. It can't bring the knowledge that I have. So that is the ultimate also problem that we see on the real ground. Any questions here on the challenges part?
No, ma'am. In chat box, there are no questions.
Madam, am I audible?
Yes, sir, you are.
Actually, there is a big chances of bias, data bias with the human being, but the computers are devoid of emotions. We can expect more accuracy when compared to human being bias. So you said that 90% is huge enough to take 80% is expected. So why can't we make it this? Because that is, they don't have any emotion, so they will take without bias.
See, that's one side of the problem, sir. They don't have bias, so they will only look at the parameters and they will make a decision. But if you are a human, you may have a bias, but you can see beyond what the computer sees, right? So I mean, let's say, if you teach a computer that, a person who comes with a cold, cough, blah 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 blah, he is having pneumonia, just a simple, so it will only look at pneumonia. But when a human or a human doctor looks at it, he might evaluate some other angles which may have probably a bigger problem than pneumonia or a smaller problem pneumonia. Because if you leave the emotions aside, our analytical capabilities are better. I would not say better, are beyond what a computer can think, because a computer can only think to the extent that we train it. So unless there is a way I can put 100% of my brain into the computer cannot think like me, right? So that is where the challenge is happening. And if what you said is right, that in fact there have been some applications that have been invented in the US, and their cancer determination precision is higher than that of the doctors. I mean, the devices are detecting cancer. I mean, the device has probably higher precision than a doctor, because what happens is when a doctor does a test, he'll take a small piece for biopsy. Under that, they will, probably take only one small millimeter for looking at the cells. Under that, they will look at only some random cells and say, okay, these are either cancerous cells or non-cancerous cells. When an AI machine looks at it, it can run that analytics, that thought process on the entire set of cells. In that 1 inch piece of biopsy, if there are, let's say, 10,000 cells, the AI machine will not take time to be waiting for one cell, right? It can run that query on all the 10 lakh cells and say, okay, yes, there are cancerous cells, 1 or 2. For the doctor, it is a higher probability of missing out than what an AI machine can do. But still, the AI machine can only do what it has been taught. So that's what I said. The trust issues and data bias is very important, because it will only think what you teach it. If you don't teach something or you miss teaching it something, that part is gone. It will not use that part because it doesn't know it. That is the gap when you compare it with a human being.
Thank you, madam.
Any other questions?
Yes, ma'am. There is a question in the chat box. Which are the projects that students of computers can do as their mega project, which are very helpful for the society?
Okay. So see, there are many they can do for the society. I'll probably give you some smaller examples. Okay. Now, in the AI space, there are small softwares that are available. What you can do is that softwares, when connected to plants, they can tell you, whether the plant has is getting the right nutrition, has got enough water, etc. So it can actually give you an input on the plant growth. And based on that, you can determine what nutrition, additional supplements have to be given to the plant, how often should you water the plant. And this is how they are helping villagers in the farming area. Just by some AI and IoT driven tools that are being integrated into the plant. When I say integrated, they are reading the parameters from the plant. And then it is helping, farmers to determine how much nutrition should be given, how much water should be given. Similarly, the soil testing that is being done, using AI, it is helping determine which plants can be grown very efficiently in that area. That is small things. And then, you can you can do some medical imaging related things, and you can help doctors read the reports faster, or based on some parameters, help build tools which can help in the doctor to diagnose faster. Like, now with all these remote things, the doctor cannot hold the pulse of the patient, right? So you can build small projects or small devices which the patient can, put it on their machine. So it'll read your pulse, it'll read your BP, it'll probably read your blood pressure, your oxygen monitoring, etc., and communicate to the doctor. So that's a very simple tool. And I was reading the other day, there are apps that are being built which can, get your oxygen reading. So if you can build an app which can get your oxygen reading, your pulse, and it can communicate to the doctor. So the things that, you go to a doctor's office and have to do don't have to be done. And there are apps which can read your body mass index, etc. If you feel, give your weight and height, etc. So these are some common life examples in which we can help the society. Small things, but making some impact back to the society. Or you could, school library, because if you talk about Andhra University, it has a big library. You could use AI to determine how to store or put some IoT devices to track the books in the library, etc. You can do that. So because see, I maybe saw a lot of books get lost here and there in a big university like AU. So you could do some things like that also.
Ma'am, there is another question in the chat box.
Yes, ma'am.
Ma'am, can you suggest a few online courses that we can take to learn about machine learning and deep learning?
Okay, so this is a question I'll park for now. What I'll do is I'll respond to Dr. Krishna Manjari on this, and she'll send that to you people. Is that fine?
Sure, ma'am.
Yeah, I'll take this as an action, and I'll come back to Dr. Krishna Manjari on this.
Please
Any
go
other
ahead,
question?
ma'am.
Yeah. So now we've talked about multiple use cases. We've talked about multiple success factors. Sorry, challenges. Now let's see, what is it that can be done or needs to be done to make AI a success. So we will look at it from 4 perspectives. One is the people, process, technology, and data. So we are giving it 4 dimensions. So now what is it about people? So we have to educate the people and empower them. So if I want to bring an AI machine, or an AI application for clinical trials, then I have to get the investigators on my side. I have to get the clinicians, clinical, doctors on my side, and explain them how this AI is going to help them and empower them. So, help them and take their view, do an inclusive part. So, educating and empowering people, that giving them the comfort that on a shop floor, giving them the comfort that they're not losing their jobs, or or they will be given training, given time enough to pick up and learn the new technology that is coming. So that kind of things, an all inclusive approach will help in bringing people on board. And also embracing change, making that whole change management process, taking the team along with you, because that is very, very important. Unless you carry the people along, no project can be a success. So doing a change management, educating people as to why this is needed, what are the benefits, how should how can we work with this together? We are not saying either or. We are not saying either the technicians or the AI machine. We are saying let's take along AI. So it's both. It's not an either or option. It's a both option. So those kind of knowledge, empowering to the people, explaining them, taking them together will help in addressing the people factor. Now coming to the process factor, since you have to define processes in the organization for any AI implementation, especially the regulatory parts, because if the AI machine is making a decision, then I have to say what should my training data be looking like, what are my guidelines, what is my percentage of accuracy that I'm expecting, and then if I'm deploying an AI machine, how am I going to make sure that there are some quality control checks beyond what the AI machine is doing. So I have to build in quality checks. Say, for every 10 records that the AI machine is processing, I'll have 1 record verified by the human that it is correct. So that one is anyways randomly, or you could make a risk-based choice that I'll probably have the machine process the record, but the risky, the ones which have a high risk parameters, those will be verified by a human alongside a AI. So you have to put the processes in place into how the AI machines can be used given the circumstances. So the entire ecosystem for the adoption has to be built in by putting in processes in place. And then Also putting in the compliance, looking at the compliance requirements. FDA or the CDSCO in India, not the or the ICMR, they may ask for your evidences saying that, okay, how do you know that your AI machine is working correctly? So we have to put in the compliance requirements, also ensure that, especially for wherever there is a human decision making involving patient safety, product quality, or data integrity, we have to make sure that compliance requirements are met from a compliance perspective also. So that entire ecosystem of processes has to be laid out. There will be changes, as we keep learning the new AI systems, we may have to tweak the ecosystem, but you have to have some foundation in place to have to start going into an AI direction, getting into the technology. So we spoke about it as one of the challenges in the previous slide, that technology has to be upgraded. You have to bring in new infrastructure, new applications, invest in upskilling of the people. So not upskilling of your technical people, the pharma company, and if you're hiring an IT service provider for doing the implementation, the IT companies also have to upskill, because IT cannot work in isolation, right? It's IT and business have to go together. If there is no business, IT doesn't exist. But at the same time, business cannot, if IT has to succeed, business has to go together with IT. So both sides have to make sure that their people are upskilled to the latest technology to be able to implement and use both ways, from an implementation standpoint and from a usage standpoint also. And last but not the least, data. So we talked about the data, that data should be organized. Even if it is unstructured, it is fine, but we need to have them in one place where from we can read them, understand them, organize them in the required format, and then agree on some standard formats across the organizations, or at least inter-organization also, and do make sure you define it. The data format, I'll just give you one very, very, very simple and naive example. Okay, you go to any pharma company, I mean, even looking at the name of a person. At some places, it will be written in short form. In some places, it will be written full form. And then, some places, the date of birth will be written in a different format. Some guy will write it in a different format. So if all this is in a computer, forget between organizations. In an organization, between departments, if you don't have a format standardized, there is no way you will be able to use the data. So say I write the data as 6th March. So I'll write 06 March. Some guy writes it 0603. Some guy writes it in an American format in 0306. That's sufficient to confuse and not make the data unusable. So data organization, format, standardization, interoperability, and that is what the whole concept of master data has picked up. You pick up the data from multiple sources and then align it with the master data. So irrespective of how it has been written somewhere, the system can conceptualize it and say, okay, it's the same thing. I mean, it is still 6th March, irrespective of how you have written it. So making the data usable and having the data in the correct data and usability, both also make a lot of are very, very critical for the successful implementation of any AI application or AI project. So that's 4 dimensions to bring in success. Any question?
Ma'am, there is a query.
Yes, ma'am.
Satyanarayana, what are the challenges faced during retraining AI algorithm?
Retraining AI algorithm, yeah. See, retraining an AI algorithm is almost as good an effort as training an AI algorithm, because the minute you are retraining, what you are doing is, see, retraining in the simplest words I would say is undoing what a person knows and making them learn something new. So you have to put this same effort in retraining the algorithm, because your data set is changing. So you are enhancing your data set. The effort is the same. You have to test it with that the AI tool is actually able to understand the training data correctly. So retraining is almost the same effort as training of the algorithm, and that is why AI algorithms are one more challenge. As long as they are self-learning or retrained, they consume a lot of effort from the IT and business team to bring them to usage. So it's almost as good as training a new algorithm.
Ma'am?
Yeah.
Sukhwan Singh ji, do you have any query? You raised your hand. He raised his hand. I don't know, ma'am.
Maybe he has again some some challenges with respect to his technology. He had logged in from a mobile, right?
Okay, ma'am. If he posts his question, I'll be asking you.
Yeah.
Kindly please go ahead with your presentation.
Yeah.
Okay, ma'am. I'm audible now.
Yeah. Yes, sir. You are audible now.
Sorry, ma'am. Actually, I'm logging in from laptop as well as mobile because of this audio issue. Ma'am, if you're through with your presentation, I would just like to ask a question, which you may find a bit childish. So I'm sorry for that beforehand only. When this AI and ML are all combined together, do you think that the problem that has been shown in the various movies like Terminator, they're likely to come true?
No, can you please repeat the last part of the statement?
Yeah, ma'am. We all have seen the movies like the Terminator, if you remember, Rise of Machines.
Yeah.
So, is anything like that a real possibility?
Yeah, maybe 50 years down the line, or I don't know, if we have seen the last 2 decades a huge transformation from where we were. I mean, 2000s we didn't have mobile phones, and today we don't really need a camera, don't need a watch, don't need a laptop. We are just surviving on mobile phones. We're doing everything. So maybe down the line we will see a day where there will be personal drones, like we have personal mobiles. We may have personal drones for each of us. Yeah, I don't think the day is very far off. Maybe another 2 to 3 decades maximum, we will see those days as well.
And ma'am, then don't you think that it will be a threat for the survival of humanity?
The survival of the fittest, Darwin's theory. So I think that's what.
Maybe we should put up some. Maybe we should put up some quality checks in our AI and ML protocols. Then
yes, yeah, we have to have quality checks, because I mean, at the end of the day, the control should be with the human. That as long as we can ensure that things are there. But see, if you see today, Sukhwan Singh ji, mobiles are controlling us. Whether you agree to it, they may not be controlling us in a literal sense, but they are literally controlling us, if not in a literal, in a other sense, because we can't live without a mobile. People are hooked to the mobiles all through the day. So I mean, they are eating up our entire time, and that's one way of controlling us, right? We thought we control mobiles, but if you look in a I would say philosophical sense, the mobiles are controlling humans today.
Thank you, ma'am.
Yeah. So,
thank you, ma'am.
So I don't see that day much far away. So that's pretty much all I had. So if you all have any questions, I can try to answer.
I request the participants, if you have any queries, please clarify with ma'am. Ma'am, there is a question in the chat box. May I?
Yeah.
Ma'am, what are the limitations of AI?
The only limitation of AI is understanding the human brain. The day a human brain can be translated into AI, there will be no limitations. I mean, if Tesla can create a driverless car using AI, I don't see there are many limitations out there. It is just that the human mind, the day it gets translated into a computer, the processing of the human mind, there will be no limitations left.
Thank you.
I think that is the day, Sukhwan Singh ji, what you asked will happen.
I hope that day actually don't come, madam.
Yeah. So other than that, I mean, it is just about, translating whatever is the thought in the human mind, the way we operate. If you can translate that into the machine will do it.
Yes, ma'am. Of course, it's a very great help for as far as healthcare is concerned, because controlling insulin delivery into a person who is type 1 diabetic,
Yeah.
or at the same time controlling vitals of a patient who is in NICU and not being attended by anybody. Of
Yeah.
course, AI and ML are going to help. But
Yeah.
again, as I said, my fears.
Yeah. So fears. See, that's everything. The nuclear bombs. People have used it for generating electricity. People have used it for bombing Hiroshima and Nagasaki. So anything drones, people have been using it for surveillance. Our neighbor has been using it for sending arms and drugs to our country. So it's about how you use it, and it's all for the depending on the human brain. Finally, at the end of the day, how we use anything for good, for bad, is driven by us. And yeah, just because you raised about this about there is a device that is there in the market. I think it's called iCare. I forgot the name. Yeah, it's iCare or iBeat. So it is a device, and what it does is, if you wear it on your machine hand, it will monitor your pulse, your BP, and your cardiac rhythms, etc. And if it finds any symptoms where your see the some protein is there, right, for heartbeat, thrombo something, I forgot the name. I'm not a pharma student, so it will look at that protein levels in your cells, and it can detect heart attacks before and notify your physician that this person has a possibility of having a heart attack. So even Abbott has softwares which can help the doctors in the hospitals identify if this person has a possibility of having a heart attack, the near possibility, not the far possibility. So those kind of devices are also coming into the market, and people are using it.
Thank you, ma'am.
Yeah. So that's all I had from my end. So any questions, if you have, you can post, or else Dr. Manjari has my contacts. So if there are any further questions, she can always send that information to me, or connect you with me. And I would like to thank you all for being patiently, interacting with during the session, because that's more important. Otherwise, it sounds like a very boring one-way preaching session. So really thank you for showing the inquisitiveness to ask and get new things. And I'll come back to Dr. Manjari on the trainings for AIML that you people asked.
Thank you, ma'am. May I ask you to close your session, PPT, please?
Yeah, I'm doing that. And I'm stopping the screen share.
Yes, ma'am.
Yeah.
Dear Madam, Miss Alaya Somayajula Naga Kavita Garu, we are very glad to hear about the challenges for the adoption. Your valuable talk helped us to enlighten our knowledge and was very, very fruitful to all the participants. In your lecture, you explained various aspects and challenges of artificial intelligence. Various stages from identifying disease to post-marketing surveillance in pharma value chain in pharmaceutical industry. Applications of artificial intelligence in healthcare management. Ma'am, you have a good visual in the illustration regarding the management of Parkinson's disease with the artificial intelligence in the beginning. So you have discussed about drug discovery and development, like speeding up the discovery during preclinical trials, creating cure for complex and rare diseases. You spoke about evaluating clinical trial data and run better analytics. You spoke about quality control and predictive maintenance, pharmacovigilance, monitoring drug adherence and dosage, diseases identification in patients, epidemic prevention, manufacturing, sales and marketing, surveillance where artificial intelligence is used in pharmaceutical industries are clearly demonstrated by you. Various key success factors of artificial intelligence, like success factors including people, process, data and technology, You cleared us very well, ma'am. And you spoke about the challenges of AI, technology challenges, skill set challenges, regulatory challenges, trust challenges, data quality, data security and privacy, data bias challenges, change management, and ultimately AI solution. You gave an excellent presentation, ma'am. With your lecture, we have gained an ample amount of knowledge. Thank you for sharing your knowledge and spending your valuable time in clarifying our queries very patiently and enlightening our participants. You have taken all of our heart, mind, and soul into your control throughout the session, and involvement of delegates throughout the session was so attentive. We are so proud of you to have you for this session. I hope all the participants have thoroughly enjoyed, and you are the one and only woman for this FDP program. So I appreciate for that also. And thank you so much, ma'am. Have a nice day.
Thank you, ma'am, for this opportunity. It feels good to be back, going back to your university and giving something back to the university. So it's my pleasure too.
Yes, ma'am. Namaste. Thank you so much.
Thank you.
I now hand over the mic to technical session people.
Yes, ma'am. So can we conclude the session, ma'am?
Yes, you give the general instructions to the participants.
Yes, ma'am. Dear participants, kindly note the instructions during the sessions. All participants, if you can pin to the screen the presentation, you can view the presentation in a full screen mode from your device. And all participants kindly post your technical issues, if any, in the chat box to avoid interruption to speakers and other participants. Whereas for discussion, you can unmute yourself and talk to speaker and clarify your doubts. If not, you can post your queries related to the session in chat box so that at the end of the session, we would read out the questions to speaker for clarification. We have allotted about a 15-minute duration to open up for discussion, and you can please unmute yourself and you can be active in the sessions. I now request all the participants to come to the video mode so that we can end up the session. Ma'am, can I proceed?
Yes, please.
Sai Padmini ma'am, Ankita ma'am, Dr. Jagadish sir. And here we go. 3, 2, 1. Thank you.
Jagadish Garu, please come in. Technical team, kindly wait for sir.
Yes,
let's have one more pick with. I request others to please join in video mode to have a pick.
And here we go. 3, 2, 1. Thank you, everyone.
Thank you, technical team.
Yes, ma'am.
Dear delegates, we'll be having our session at sharp 3 o'clock. Introduction to Artificial Intelligence and Machine Learning related to Pharma Industry by Rajneesh Vasanta, CEO, Antar IoT. I request all of you to join the session before the 10 minutes of the schedule. I request all of you to leave the session and go for the lunch break. We again join at 10 to 3 for the third session of the second day faculty development program. You can leave the session and rejoin the session at 2:50. I request all of you to leave the session. We will be rejoining the session at 2:50 p.m. Technical team, you can close the session.
Yes, ma'am.